Yeah,
So
here's a true fisher. We need to wait till we get the stream going.
yeah, let's see.
We're live. We are live, so welcome back everyone. We'll start with this morning with another three set of presentations. We start with Samuel Gibson presenting us with his thesis work.
Indeed. All right. Hello, everyone. Those who don't know me, I'm Sam Gibson, and today I will be presenting my thesis, which is mapping and measuring mangrove restoration with carbon sequestration in Sankandi, the Gambia. So, brief background is obviously mangroves are a salt-tolerant species that are found in the tropical and subtropical regions of the world, and they provide a vast array of socio-economic and ecosystem benefits. Some of these might be nurseries for fish, physical use for like construction and firewood. They also have spiritual links, such as in Sankandi, and of course they can provide green tourism, with many people going to see them for their birds, such as twitchers. Another key aspect of mangroves is they are recently discovered to obviously be great carbon sinks and provide a large amount of carbon sequestration within the biomass, but primarily within the sediment. Even though they are such an important part of the ecosystem, there's been large causes of degradation over the last few centuries, with 50% loss since the industrial era. And again, anthropogenic reasons are the main cause of loss here, with overexploitation and the destruction for land clearance in coastal regions. With their importance in carbon sequestration, there's large drives of initiatives with governmental and organizational levels. Governments like Indonesia have planned to restore 600,000 hectares of mangroves. And you have organizations such as the Red Plus program, with businesses using these models to have funding for green restoration in rural areas such as Sankandi and such other parts of the world. So more local context. So Sankandi is in the Gambia, which is on the west coast of Africa. It's about 200 kilometers inland from the sea, and although it's so far in, the tributary from the Gambia, it is still tidal, so it is salt water that the mangroves are experiencing. The main loss that occurred in Sankandi specifically was between the 1960s and 1980s with the Sahelian drought. This was also exacerbated by continued exploitation by the locals with firewood and construction, as in this area, mangroves are predominantly used for their construction of homes. This has led to the loss of fish stocks and loss of arable land with increased salinization spreading out from the tributary. And with the rural community at Sankandi bringing primarily subsistence farming to have their income, this has been a severe hit to the locals in the area. So because of this, a charity called Sankandi Youth Development Association has had some success with initial planting starting in 2012, with each consecutive year getting more and more funding from local charities to plant more and more mangroves along the degraded land of the Bintabalong tributary. These plantings have predominantly been, in fact entirely been Rhizophora mango, which is native to the area. Sankandi Youth Development Association have planning to do future work with a Scottish charity called ASAS, which is where my professor linked me to. And they're going to try and do some green carbon credits in the future. So this is why my pilot project is here, to try and work out how much carbon sequestration is occurring in Sankandi and see potentially the viable incomes that they could get. They've also discussed the moving to a more salt-tolerant species of Abyssinia gemminans, as most of the land that Rhizophora could thrive in has already been planted. So they need to expand the types of mangroves to get more mangrove restoration involved. So the aims and objectives of my project, so my primary aim was to do a mapping surveillance of the region to get a high detailed resolution map of Sankandi land, and then delineate the land functions, so the land uses, and the different ages of the plantings. We also wanted to do some pore water gradients to see how the changes of salinity occur across the site. So this can help facilitate the decision to use Rhizophora or Abyssinia. And finally, an additional aim was to do carbon sequestration to get an approximate idea of the changes in carbon sequestered in the sediment, and then to have some rough estimates to see how much Sankandi land could potentially generate in carbon credits. So the fieldwork itself So the first part was droning. So I just droned at about 150 meters above sea level, between 9 and 10, to keep the light constant. And I did a square zigzag formation across the site until it was complete. This took about a month to do. For the pore water transects, they were all perpendicular, randomly selected across the site. And to gain an idea of the changes in salinity in the pore water, I dug up to half a meter. To the furthest extent that I could dig, because in the summer heat of Gambia, the ground hardens very quickly and you can't dig any further. And I always did this after high tide so that I could feasibly dig in the land. The carbon biomass was just done with allometric equations from Komiama and Langefeld. And these were done with measurements taken at breast height at 1 meter 30, and then when it wasn't possible because of the size of the mangroves, at 30 centimeters. These were done with 20 randomly selected trees in each of the plots, and I'll show you the plots later. For the carbon, in the same plots as the trees, 5 random samples of the surface sediment were taken, and these were then dried and taken back for lab analysis in Edinburgh. So for the lab work, 2008 individual photos were homogenized to make the restoration site map. This is using software from Drone Deploy. And then afterwards, slight delineation was done by prior experience in the area, talking with locals, and looking at the maps. For the carbon biomass, these are the allometric equations used. But as you can see, there is no allometric equation for the 8 mangroves aged 2 to 4. So for this, I had to approximate it using the ratio seen between these two changes in biomass for the 2 to 4, and I'll show on the graph later how that looks. And for the carbon, this was basically just using muffle furnace to do loss on ignition. And then from here, you can approximate the loss of carbon in the sediment. So, the rocks. This is what I'm very proud of. This beautiful map of Sankandi restoration site. So this is the entire area here that Sankandi owns. And the way land ownership works in the Gambia is each of the local regions, tribes, has a particular part of the river surrounding. However, even with this, local tribes or other communities will often plant on different people's lands when they have the funding, say, get paid by these green initiatives to plant mangroves, so they just start planting. This can cause later issues when it comes to ownership of the rights of the carbon credits, which I'll discuss later. So, to site delineation from the maps, I came up with 4 basic land functions: plant and established. These are regions of the restored site that have either planted mangroves or naturally occurring ones. I couldn't distinguish the entire site between who owned the rights to it, either the locals in Sankandi or other tribes and other communities in the area. So that's why they're a whole collective. We have potential spots for Abyssinia and Rhizophora. Rhizophora had been primarily taken up, so there's only a small area of potential Rhizophora. That's why they're planning to move to Abyssinia, which has a much greater area of potential expansion. And we have the mudflats. These are areas that apparently historically used to have mangroves, but now, because of degradation, have gone, and they have become incredibly saline. So these could potentially be used in the future with extensive work, such as digging canals and other things, to establish mangroves there in the future. So these are the planted regions that we know exactly the age they were planted due to photo evidence. For my experiment, I took these 4 plots, which was the 2016, which is a small group here, 2018, which is just behind it, 2020, which is here, and then the most largest, which was 2022, so this. There are other known planted ages of these ones here, but because of the limitation of bringing back sediment from Gambia, I can only pick 4. And of course, the potential site here. Oh, these are potential for Rhizophora, I should say. So, just a quick look at the salinity. Unfortunately, you can't see the transect lines on the map, but we had ranges of, so the source water had about salinities of maybe 37 to 40 parts per thousand, and within the Rhizophora plantings, the ones that have been historically done, these would roughly go up to maybe 60 parts per thousand. However, in the areas which are designated for Abyssinia expansion, these can go up significantly, up to, in some regions, 80 parts per thousand, which is very saline, even for the saline-tolerant species of Abyssinia. Here we have the changes in carbon stored. So this is the results from the loss on ignition. So this isn't a direct image of the loss of carbon. However, with the average approximation of 50% of the loss of ignition being carbon, we can come up with some ideas of how much of it was actually lost. And for the biomass from the mangroves, these are the allometric equations results. And in red, these are the ones I had to approximate because there were no allometric equations for the mangroves aged 2 to 4. So by combining these two, we can come up with an average carbon change, which leads to a trend of about 5.6 megagrams of carbon annually sequestered across the site. By converting this into CO2, we can then get some approximations of how much CO2 is sequestered, and then with the going price that the ASOS, which is what Sancandy is trying to link with their charity, being $20 per megaton, we come with an average price per hectare of sequestered carbon or carbon. For the rest of the project, I just used the conservative value of 230 to be realistic about how much the community could potentially get if this was to go ahead in the future. So with a conservative estimate of the tested land I've done, which was added up to be about 10.38 hectares, Sancandy could generate roughly $2,500 annually. So in conclusion, we have so the establishment of the baseline map. So in conclusion, we have the establishment of the base map, which is then used for current and future planned restoration initiatives. With talking with the locals, we can delineate the current land use. And from past photos and talking to the locals, we could work out the average planting ages of different plots. With the salinity, there is ability to gain greater understanding of the changes across the site, and this can help facilitate future plantings and ideas of which mangroves to plant, and of course, the calculations of the approximation of carbon. The potential revenue, so this is if of all of the known planted sites by Sancandy directly, sums to about 5,500 per year. If over the entire planted site, so this is also all of the planted mangroves, if it turns out that the rights are by the owners of the land, not those who planted, they could potentially generate 33,000. And across the entire site, without the mudflats, this is with the potential of use of Abyssinia and Rhizophora, further expansions, it may be 37,000 annually. So for a rural community such as Sancandy, this is a significant amount of money which could help future projects in the area and help with general infrastructure and life betterment. So for future work, so obviously a lot of my work was approximations and rough estimates. So ideally, with the loss on emission, you would do a CNN analysis to work out how much of the loss on emissions was carbon, other than just taking a generic standard of 50%. Again, as I've stated, the clear understanding of the ownership of the land is very important because there's not direct legislation in Gambia with the ownership of the rights. It is more just done on word of mouth. This is owned by Sancandy. This border is up to the tree. So there needs to be greater levels of legislation or understanding to know who owns what part, and even then who's planted on it, who gets the right to the carbon. And this is the time of the plantings I was in German. So when I was over there in Sancandy, it was in the middle of the summer. So the mud flats had already had a lot of levels of evaporation. So the mud flats themselves were incredibly saline. Any attempts to do restoration with Abyssinia, even though it is a more salt-tolerant species, would end in greater mortality than potentially in the wet season, or if there was further work to help desalinize it before the planting. So, and it's also important to mention that although there's potential future incomes from carbon credits, these schemes can take many years to come into fruition. You know, the community were talking with the local charity, and this was looking to maybe 2027 at the earliest, most likely 2030. So it's important for the communities that plant these mangroves not to be doing it directly for the carbon credits, but also extra facilitation. So from this, they hope to gain reduced salinization so that they can start re-farming the arable land surrounding the area. But it's also important that potentially they can use the stuff they've already started established for additional green income. This can be done through green tourism. And of course, in Sancandy, they're using some of the money they get from the projects they're getting to plant beehives to help get another level of income for the community, and also to help facilitate the pollination of these mangroves. Yes, I went quite quickly, but thank you everyone, and yeah, hope you enjoyed.
Well, thank you, Sam. You used 14 of your 15 minutes. You might have taken the time to speak a little bit slowly and breathe a bit more, but it was fine, I think. So I'm going to ask the members of the jury to start the discussion. Who wants to go first?
Sure.
Mm-hmm.
I was wondering about the current use of unvegetated areas, although it's super saline, and famous to well, are they used by the locals for some reason?
No, so in the area of, so surrounding Sankandi, Sankandi themselves aren't using the saline areas because it's too saline for them. However, in neighbouring communities, they actually don't want to restore the mangroves because they're using it to collect salt. And they've been using these areas to generate income by collecting salt, sorting it out and selling it. However, Sankandi, actually while I was over there, there was a few days we couldn't go down to the Bintabulong area because of cultural events where we weren't allowed to see. So for them, the mangroves have been very culturally important. So they're wanting to restore it, not only necessarily for the desalinization of the land, so they've used it for arable, but to get back some of what they've used historically for cultural significance.
Okay, okay. I'm not very familiar with a carbon budget system. How can they extract? How can they get money from this?
So basically, if you have a baseline of how much carbon is already in the sediment without any mangroves, and you can see the trend of sequestration, you can have you can approximate an annual sequestering of carbon through the biomass and in sediment. So this change, so the percentage, the amount change per year, they can say, well, this is the amount of carbon we're taking from the atmosphere, putting in the ground, so we can then sell these as carbon stored.
From who?
From who? From communities, so like for businesses that want to kind of be, start being green by offsetting the carbon that they are producing.
So industries in Europe and the US.
Yes. So basically,
keep emitting carbon because people in Gambia are planting trees.
Essentially, well, they're going to be emitting carbon either way. At least this way, that rural communities such as Sankandi can generate some, and they can actually get finance to start planting the mangroves. And especially with ASAS, so the charity that is working with the community at Sankandi, their aim is to only be working with companies that are actively trying to reduce their carbon impact and are just emitting for now. They don't want to work with people who are just greenwashing, saying, oh, I can pollute as much as I want because I'm paying people to plant.
Okay, and regarding the land ownership, how does it work there?
So, up to my understanding, whenever I spoke to the locals, it was always a little bit iffy. So, in the Gambia, obviously the Gambia is a very thin country. It is all based about, I think, a couple kilometres either way of the Gambia River itself. And so the communities have all been allocated particular parts and access to parts of the tributaries. So the communities themselves, at least word of mouth and their understanding, have different segments. However, that does not stop other communities, when they get funding to plant mangroves, to do anything, going into their land and start planting the mangroves. So the land ownership, this is what it needs to be really for future projects set in stone, like who owns what and by what degree. That needs to take place. So it's not as clear cut as it would be in other countries.
Okay.
You can more time.
Yeah, maybe we can come back.
Yeah, what's a part of the carbon which is actually sequestrated from the absorption of CO2 by photosynthesis, but it's a part of this that is actually sequestrated by the mangroves? And more generally, what is your definition of sequestration of carbon here?
Okay, so when you talk about carbon sequestered by the mangroves, are you talking about the biomass or what they put in the sediment?
In general.
So the ratios across the site, so with mangroves planted, two-year-old mangroves, about 1% of the carbon in the hectare of land is in the biomass, and after 8 years, 10% is in the biomass, and the rest is in the sediment. And so what I've gone for with the carbon sequestered overall is just the changes in carbon that you see across these different plots.
Okay. So here it's the net absorption of carbon you're talking about.
Yeah.
And also, more generally, what are the additions of a drone method that, like yours, compared to a satellite method in terms of accuracy? And could you use both methods to also take advantage of all the sensors you could have on satellites? For
sure. So when I compared my drone footage to satellite imagery, like from Google Earth and stuff, it's the level of accuracy that a drone can do is it can reduce the age of plantations. So like the plantings. So with a drone, you can see, I think, up to, so when a 4-year-old mangrove, you can see it with a drone, but you can't see it with a satellite. So this is actually very important when it comes to large-scale mapping of mangroves with satellites. They often don't pick up young mangroves, and they'll only pick up the fully established mangroves. So with drone footage, you can pick up a much younger age of mangroves, which can then help get a better understanding of the restoration initiatives that are happening in these areas.
Okay, that's for direct observation, but
Yeah.
couldn't you use also the sensors from satellite, not the light we can see, but other sensors that you could use to track other variables?
So like RGB, sorry, not the light. So I think, what was I trying to say? From my understanding, you can definitely, I was looking at other projects done in the Philippines with drones, and although you can use satellites and other aspects to pick up different areas of mangroves, with in situ footage, what I'm trying to say. So you can for sure. But I can't think of the individual right now, the different specifications.
Talking pigments, also tracking carbon absorption from satellite and these kind of things. I don't know if you could.
I don't know about that.
Okay.
I don't know about that.
I can check.
Yeah. There's this relationship that you're showing here with adjustment to the model that is not amazing and a non-significant relationship, which would explain like 65% of total variance.
Mm-hmm.
What other variables can potentially explain the relationship between the carbon stored in sediments and mangrove?
So like
You consider here the age of the mangrove plots?
Yeah.
What other variables can be relevant here? To explain carbon sequestration.
it depends. So it could be distance from the main source of the tributary.
Yeah.
That can play a factor.
Did you explore this?
So with the sites, so these were the measurements taken. These were the age plots. So this is the main tributary.
Yeah.
So this main tributary, this is the Binge-Belong tributary, and all of the planted rhizophora currently are on the tributary, which is along here. So from here at least, potentially variance in carbon could be done by the scale of the size of the water source that it's near.
Mm-hmm.
It could also be dependent, could be the length of time of inundation. So obviously some areas, such as up here, would be underwater less, so that means it's probably going to be more aerobic respiration than in areas such as here, which are inundated longer.
Okay. I didn't see anything regarding the density of mangroves.
No, so
From the few photos I saw, and also maybe was it the same area where Fantine did this?
yeah.
But it's kind of heterogeneous with some areas of quite high densities and some other individuals scattered here and there.
So for these age plots, if I remember correctly, the densities ranged from 1.7 mangroves per meter squared to 2.5.
Okay.
In these tested plots.
Okay.
Yeah, and these are all rhizophora.
And did you compare your carbon sequestration estimates with data from the researcher? And there's lots of data for both Abyssinia and
So for sure.
Yeah.
So I mainly compared it with work in Senegal, obviously the surrounding region of Gambia, and what I found was conditions were very similar in terms of salinity, composition of sediment, and mangrove species. However, Sankandi, so this area seems to have a higher level of carbon sequestration, I think by about 20% compared to what was in surrounding Senegal. I think this can be understood by the rural nature and the fairly rudimentary science done for this. So using loss of emission instead of CNN analysis could provide the reason why there's such a disparity, well not such disparity, about 20% between local regions, which is similar.
20% is not too bad.
Yeah. Okay. Yeah.
Okay. Thank you.
Yeah.
Well, at the end, in your conclusion, you were mentioning other sources of income, so I believe you are not relying only on carbon money to do that.
Yeah.
I was just wondering, because we're talking about $37,000 per year.
Yeah.
So that's nice, but it's
For sure.
also not so much. So I don't know if you already
Yeah.
try to calculate what would be the cost relating to creating this carbon credit cycle framework, of labeling the carbon sequestration so it can be recognized, the cost associated to the protection of the place so it's not destroyed, Because I know how many people could you hire for that?
So with St Candy, so St Candy Youth Association and Thesis, they're planning on hiring 2 people, one to oversee projects like this and other initiatives in the area, such as the beehives.
What would be the cost of that?
I'm not sure exactly, but I can't remember what did I can't I was told I can't remember.
Go ahead.
But they also are going to hire someone to do nurseries. So this is to try and facilitate and make it easier for the planting of Abyssinia. I can't remember the costs exactly, but I do recall. So the important thing with this the work is it was all community based. So when I was over there was numerous complete community meetings to discuss the importance of not cutting down the mangroves, not coming and doing what you want on the land.
And
so
that's for the labeling, because if you want this carbon to be recognized by international entities, you have to, and this is costly.
Yeah, so this will be done. This is planned to be done by the ACES charity, but I don't know the pricings of that.
But it's covered by the foundation.
Yeah, so I think that's what they'll be planning to cover it, and they take a percentage of the carbon credits. But on average, they give a lot more to the communities than other projects like Red Plus.
Yeah, we have time. No, go ahead.
So here we're heading to an active restoration process of planting.
Yep.
Have you considered, you or others considered also letting the things go and using just passive restoration? Do you think it's possible to wait and how long to have a passive restoration?
I think with so with Abyssinia, so I was going through the Google Maps images and seeing about 2010. This was completely desolate. This was just completely all dead, and this was in 2010. So these Abyssinia mangroves, they've come there naturally, so they are occurring. But the reason why it's mainly so active and it's active restoration is because the local area, which is mainly farming with rice fields, they're having to abandon field after field because of the increased salinization. So as I was saying, it's very important for communities not just to do it, well, for just the potential of carbon, because often that falls through, but for other reasons. And so the main reason initially was to try and stop the salinization of the farmland surrounding. Yeah.
All right. Thank you very much. So if Peter wants to come and set up. One minute, so we start on time.
Sorry?
We just wait 1 minute, so we start on time. If there's ever people that want to catch you online,
yes,
there might be. All right, Peter, whenever you're ready, you have 15 minutes.
Thank you. Good morning, everyone. My name is Peter Lahif, and the title of my thesis is Quantifying the Importance of Cultivated Macroalgae and Associated Structures as Habitat for Marine Epiphytic Communities. I studied this thesis in Atlantic Technological University, Galway, and my supervisor was Dr. Jose Fariñas Franco. So, the aim of this project was to determine the habitat value of cultivated macroalgae, or seaweeds, and their ancillary structures. So, in the wild, macroalgal assemblages such as kelp forests are known to host high levels of biodiversity and abundance of organisms, and it's often seen that older and more structurally complex seaweed beds host higher levels of diversity. So our hypothesis was that more complex assemblages in a farm setting would also be associated with higher diversity. The seaweed has been farmed for a long time in Asia, and currently the macroalgal cultivation industry is expanding in the West. So if it's found to have benefits for biodiversity, it would be promising for this sector's long-term sustainability. Our samples were collected in 2018 from a site in Ventry Harbour in County Kerry, the Republic of Ireland. And at this site, two brown algal species are cultivated, the sugar kelp Saccharina latissima and winged kelp Alaria esculenta. We focused on Saccharina latissima for this study. You can see a map of the site here and the layout of the farm below. The macroalgae were grown on 1 meter long polypropylene ropes known as droppers, which were attached to floating long lines at the farm and kept suspended vertically in the water column with a small concrete weight on one end. This is a schematic produced by the Genial project, which works with algal biodiversity across several partner countries, showing the potential interactions between farmed seaweed and adjacent wild communities. These can include both positive and negative effects, such as altered hydrodynamics, input of organic matter, and potential spread of pathogens or invasive species. At the farm, the droppers are typically sterilized and then seeded with algal gametes, or propagules, prior to deployment at the site. So by the time they're introduced to the sea, the algae are already well established on them. For this study, we used both the seeded droppers as well as sterilized, unseeded ropes that would act as controls to see what kind of organisms would colonize them if the algae weren't introduced beforehand. We also collected samples in July, when algal growth was at its peak, and in October, when the algae had begun to degrade and was replaced by a different community of organisms to compare diversity between them. You can see on the top left here the sterilized droppers in a recirculating seawater tank being seeded with propagules. This is what they look like immediately prior to deployment when the algae is established. This is a dropper at the site with the Saccharina latissima growing, and that's it after being harvested in July. In the laboratory, I took the samples that had been in the freezer since 2018, and in the marine and freshwater lab in ATU, I washed them over a 500 micron sieve to extract any macroinvertebrates associated with them. The invertebrates were preserved in 70% IMS and sorted by higher taxonomic level before being identified further to species level or genus or family if species couldn't be determined. Once this had been done, the individuals of each species were counted and statistical analysis were conducted in R to determine the diversity and abundance metrics for each sample. From this graph here, you can see that in these samples from July, amphipods were extremely dominant, and in terms of abundance, they were far more than all other taxa put together, while by October, the amphipods had largely decreased and the communities were much more even, with sedentary polychaetes, decapod crustaceans, and bivalve mollusks being among the most abundant organisms. Overall, annelids, mollusks, and crustaceans were the most consistently abundant taxa across all samples. The spat of mussels, Mytilus edulus, were also highly abundant in July, since this corresponds with their spawning period, but to avoid skewing the graph even further, they were excluded, and they were also excluded from the analysis because mussels develop into reef-building organisms, they modify habitat for other species, and so they were treated more as a factor than as part of the community in this study. These bar plots were created in R to show the change in abundance, diversity, richness, and evenness between the sampling periods. So early on in July, abundance is a lot higher due to the high numbers of amphipods, and it decreases towards October. But evenness, diversity, and richness all increase as time goes on and as other species become more abundant. It was found that month was therefore a significantly correlated factor with evenness, diversity, and richness, while the seeded or unseeded treatment of algal propagules wasn't significant, and neither was the interaction between month and treatment. This non-metric multi-dimensional scaling plot was created to show the dissimilarity between different communities. The orange or red dots represent the samples taken in July. They're largely dominated by the mussel spat and by amphipods, while these blue dots represent the October samples. And these group together in two clusters. This one up here includes Cancer Gammaropsis, Pisidia, some other crabs. This one here has bristleworms, Ophiutrix, brittle stars, anemones, and so on. And there's some overlap, but these blue dots represent the seeded samples. These triangles represent the droppers with no propagules, the unseeded samples. So it looks like treatment had somewhat of an effect between the communities that developed after 6 months. Here are some of the characteristic organisms from each group of samples. And this K-dominance curve shows the most numerically dominant species between samples. Again, in July, amphipods were highly abundant. Ichthyosaurid amphipods like Jassa, Ichthyosaurus, Parajassa, and so on were very common, as were amphipods in the genus Caprella. And then with the more diverse communities in October, crabs such as Pisidia, bivalves such as Hiatella, and worms such as Spirobranchus and Polyophthalmus were highly dominant. These linear regression models were created to show the influence of muscle biomass on the rest of the community. It was found that biomass of adult muscles had a slight negative correlation with diversity and evenness of communities in October, but a positive correlation with their richness and abundance. So, time was the most relevant factor in determining the communities that developed, but it wasn't so much time itself, but rather the communities that had developed over the course of 3 months versus 6 months. The algae, Saccharina latissima, was dominant in July and hosted a community of organisms typically associated with algae, such as amphipods and errant polychaetes. But by October, the algae had largely degraded and was replaced with a bivalve-dominated community as the mussel spat grew into adults. The algae thalli mostly disappeared, but the holdfasts in many cases remained on the ropes that had been seeded, and the holdfasts provided a structurally complex habitat and an attachment point for bivalves. So it seems like, given the difference observed in October between the treatments, the presence of holdfasts had an influence on the communities of organisms present, even if it wasn't statistically as important as the period of sampling. Some organisms to note include abundant juvenile crabs of the species Cancerpagarus, the edible crab, and Pisidia longicornis, the porcelain crab, found in the samples taken in October. Cancerpagarus are fished commercially in Ireland, and if the seaweed farm is providing nursery habitat for them, it would be a promising sign, seeing as it would be producing both algal biomass and a invertebrate species that could potentially be exploited by local fisheries. Pisidia longicornis isn't consumed by humans, but it does represent a food source for fish that could be of economic and environmental importance. So, in conclusion, the farmed algae was found to harbour high levels of diversity and abundance of certain species, but it wasn't the algae itself, but rather the bivalve communities that developed alongside it on the seaweed farm that were the most diverse part of the community. This represents a potential trade-off, since farmers obviously want their seaweed crop to be in the best condition possible, and the presence of encrusting organisms such as tube worms, polychaetes, and bivalves reduces the economic value of the crop, but these are the kind of habitat-forming species that we associated with the highest diversity and abundance. One potential workaround to this is that, as with terrestrial farming in the European Union and the UK, a grant could be offered to seaweed farmers to leave aside a small percentage of their farm for nature and effectively allow part of the crop to degrade in order to improve biodiversity on their farm while still maintaining economic productivity. That's all from me. These are my references, and thank you very much for listening.
Thank you, Peter, for your presentation. You had a bit over 4 minutes left of time, so
thank you.
yeah, you could have taken more time and explained something more in detail, but we'll take that time during the questions of the jury member lecture to think
Okay.
about that. Thank you. Want to go start with
Yeah, surely. Thank you, teacher. That's a topic that really interests me. Maybe I missed it, but I didn't hear you talk about the result for natural habitats. Did you actually compare what you found with what's found on natural habitats?
Not in this study. We were only looking at the difference between the seeded versus unseeded. With the unseeded droppers had nothing attached, nothing introduced to them, so they were effectively represented the control or what would grow in this habitat in the absence of seaweed farming.
Yeah.
There are other studies going on in ATU where they are taking samples of wild algae and comparing the communities found in those to the farmed algae, but we didn't cover it in this study.
Okay, because some of your conclusions, if I'm not mistaken, you claim that the assemblages are comparable to what can be found in natural kelp forests, which I found super surprising because I don't think you can say the biodiversity that you found, either the number of species or the structure of the community can be comparable.
No, not necessarily. We did
I
find
spent a significant part of my career working on biodiversity in kelp
Yes.
forests, and even though I agree that the species are similar, I mean the diversity in kelp forest is extremely important.
Yes,
We're talking
definitely.
about hundreds of species, and I would incite you to be very cautious when you make such claims.
Yes,
I think,
yes. Maybe it's better to say that we found a promising level of biodiversity for a anthropogenic setting, but yeah, not necessarily on the same level as a wild algal community.
Okay, so that's something very, very important. And can you go back to your last slides about your suggestion of the incentive that could be offered to seaweed farmer to set aside a small proportion of the crop from nature? Can you develop on that? I'm not sure I really understand what you mean.
Well, in terrestrial farms, grants have started to be offered for farmers to effectively rewild a small percentage of the farm, say set aside 5% of the land area that would otherwise be used for crops or rearing animals, and effectively just let it go wild, allow native vegetation to establish on it, and provide habitat for insects and other organisms living there. This similar thing could be done here, where most of the seaweed crop is harvested, but a certain amount is allowed to effectively decay over the autumn period when we saw the algae begin to disappear and be replaced by bivalve communities, with those bivalves acting as nursery habitat over the winter period for crabs, amphipods, and other species.
Okay, but I think there's a major difference between agriculture on land and seaweed culture.
Yeah.
But on land, agriculture actually takes the place of natural habitats.
Yes.
And seaweed culture are on floating structures, so they don't take the place of natural habitats.
Yeah.
So if you suggest that seaweed farmers could leave some of their production for nature, you're actually introducing an extra amount of organic matter, and without any indication that it would not harm the environment.
Yes, that's true. It would increase biodiversity on the farm overall, but it might have effects on natural habitats adjacent to the seaweed farm. There have been studies done on the impact of seaweed farming on wild communities, and so far it seems like a very low impact. There's less organic matter inputs associated with them than fish farming, for example, where the fish droppings represent a nutrient input and can cause eutrophication. Seaweed so far appears to have a very minimal impact. It takes nutrients out of the water rather than introducing them, and certain studies have been done to see whether it has an effect on shading wild photosynthesizers such as algae and seagrass, but it's found a minimal effect so far. Of course, seaweed farming is still relatively small scale in the West, so as it expands in scope, there may be greater impacts, but so far it seems like it has a relatively minor effect, so that we still need to be aware of the spread of invasive species and anthropogenic activity near sensitive ecosystems.
Yeah, I agree with you on the fact that seaweed farming is probably one of the most impactless productive activity, both on land and at sea. But then why would you want to increase the biodiversity of those structures? You have structures that seemingly don't affect the environment. Why would you increase the biodiversity?
True, you might not necessarily need to. Maybe it might be best just to leave it as is. Although if there is the use of juvenile habitat or spawning habitat for fish and invertebrates may represent an additional source of income to make the farms more economically robust, although it wouldn't necessarily be of benefit to wild communities.
So you're talking about nursery, you say that you found indeed some juveniles, paggers and
Yes.
and your customers?
Yeah, so there were juvenile crabs were quite consistently found in high abundance across the October samples. They seem to favour the bivalve reefs as habitat, so it seemed like they were successfully recruiting and using this habitat as a nursery site.
I'm not surprised. Do you think this observation is enough to claim that this is a nursery habitat? And if
No,
not, if I'm asking the question that because I think it's not, what would you do if you were asked to prove the nursery role of seaweed farming?
Well, I don't think it's enough so far. It is only one study, so you need to do further work on it to be sure. If you wanted to prove that it's nursery habitat, I don't know, perhaps you would need to do a longer timescale and observe the, first of all, establish a baseline, find how many wild crabs of these species are present in the environment and how the presence of juveniles, how successfully they're reproducing in adjacent habitats, and then compare that to reproduction within the farm site to see if there's any significant difference.
Yeah, I agree. You can, if you want to talk, I have like an infinity of questions, and it's more because that's the topic I really like. It's
Thank you.
fantastic. But you can interrupt me and I can come back at them.
No, but you don't have any
Get to one, yes.
No. Just a little bit, then we come back to Gauthier.
For me, of
course.
Actually, I'm an expert here. I think too. So, well, you were trying to talk about, you were talking about this invertebrates as a possible also source of revenue for the farm.
Yes.
So how would this work? Is it done already? Where do you have some successful examples of that? Where you have both the raising of these animals and algae?
Well, there's multi-trophic aquaculture sites where organisms such as sea cucumbers, bivalves, and so on are grown alongside fish farms or seaweed, and with the nutrient inputs from fish farms, for example, those can be a source of food for detritivores like sea cucumbers, and then those can go on to be sold and generate extra income for the farm. But it might be a bit more difficult to do it with organisms that aren't technically being raised on the farm, but are simply wild organisms that are colonizing the site. But it's possible that there would be a spillover effect, similar to marine protected areas. Seaweed farms are often said to act as de facto MPAs because they exclude fishing vessels. Boats don't like to enter seaweed farms because of collisions and entanglement. So there can often be reasonably high levels, reasonably high abundance of fish and marine invertebrates within the farms that may then either produce young or migrate as adults to adjacent regions where they can be a target of fisheries.
Okay, that could be more comprehensive, larger.
Yes.
Also on a more ecological side, you were showing us with your bar plots, I think.
Yes.
That's amphipods were much really present, dominating the community in July.
Yes.
But the capods were dominating slightly the community in October, I think.
Yes.
So is it a normal ecological succession in the ecosystem? Can you explain a bit why, according to
Yes, it's
Can you just
move a little bit to the right?
this way.
Yeah, whichever.
It's ecological succession and the life history of those crustaceans. The amphipods are very much associated with kelp in the wild, and also some species like caprella are opportunistic. They're often associated with both disturbed habitats and anthropogenic habitats, and they've become a very invasive species in some regions. So in July, the community was still fairly young, and it had represented what was effectively a novel community of seaweed growing on a hard substrate. So it was overall not especially diverse, and therefore there's a high skew of organisms that are adapted to a disturbed or algal-rich habitat such as those amphipods, while the community in October had effectively undergone secondary succession and represented what we might consider similar to a wild habitat after a longer period of time. And so a greater diversity of organisms had established, and as well as that, the habitat was more suitable for species such as decapods, which favour bivalve reefs. Also, there's seasonal changes. Certain amphipods have a life cycle where they're highly abundant in spring and summer, then they reproduce, so die off over the winter. So we see a huge drop in abundance in October, and then their eggs or larvae would recruit again in the spring, and we see a peak of abundance at that time.
We have a question from Valentin.
Yes?
Thank you for the presentation, very cool.
Thank you.
I have one question. What's the lifespan of those structure? I guess that you need to change them quite often as they
Yes.
stay in water.
Yes, it's effectively.
Sorry, just second
Yeah.
question related to that. What do you think will be the effect of ending the structure on the biodiversity?
It's effectively an annual cycle. The seaweed is introduced in about March, harvested in July, and then usually wouldn't be left until October. This was just for the purpose of this study. It was left in the sea longer to see what kind of communities developed on it. So yeah, it would effectively be a spring and summer growing period, then harvested, left barren over the winter, and then the cycle would begin again in the spring. So leaving the ropes in over the winter to benefit biodiversity was what we were looking at over this study, because the longer they're there, the more organisms will settle on them, and it will continue to undergo succession until it's effectively at a stable state. If it's replaced every year, it will effectively be reset each time to a disturbed algal community with these lower biodiversity values. So that's part of the reason we suggest leaving a small portion of the ropes in over a longer period, while the rest of the crop effectively undergoes turnover and is reset back to the early stage succession each year.
Okay, thank you.
I have a question before we go back to Gauthier.
Yes.
Some of your initial hypotheses were based on the relationship between complexity of the habitat and diversity.
Yes.
My next question.
That's kind of my topic right now. But I'm sure you'll have more to say about it. I'm wondering, so your initial hypothesis was basically if the seaweed is older, it's going to be more intricate, it's going to be more complex. But then also in your work, you have this, you show as well, we also have the bivalves that are habitat forming, and they're offering higher diversity in terms of alpha diversity. Can you compare the complexity of these habitats? Have you thought of any way of quantifying, measuring the complexity? And if so, is that something that you think is I don't expect you to have the definitive answer, of course. Do you think that it would be possible for us to quantify complexity in any kind of biogenic habitat like that? And do you think that would be important to be able to really understand the relationship?
I have seen some studies comparing diversity across biogenic habitats. There are different communities associated with bivalve habitats versus algal habitats, which makes it a bit trickier because bivalves are a hard substrate. They're better for encrusting organisms like tube worms, while algae are a bit more difficult to settle on, and they tend to have more mobile fauna like errant polychaetes and amphipods associated with them, which means that the comparisons aren't one-to-one, so they're not all that informative. But you can look at overall species abundances and productivity between them. And yes, we did talk about more in the wild, more older and more structurally complex algae are more diverse. So we weren't able to look at that over this study, because even if the algae isn't harvested, by the time winter arrives in Ireland, there's usually strong storms off the west coast, and that will strip the algal fronds off of the farm longlines. So even if, in the absence of harvesting, they don't tend to persist for as long as a wild algal community would, which is why we focus more on the ecological succession to bivalves. So yes, to answer your question, you could compare them, but you would need to take into account the different communities present in each kind of habitat.
All right, thank you.
That's it.
Yeah, you didn't say anything about the seaweeds that colonized your ropes beyond the Saccharina.
Yes.
Did you observe any other seaweeds colonizing the, what do you say?
The droppers, yes. To a small extent, we did see some small red seaweeds growing there, for example, but we didn't analyse them over this study. We were just focusing on invertebrates. But perhaps some of the other researchers in ATU will incorporate that into a future study and see if there's any epiphytic algae either growing on the seaweed themselves or on the droppers that might contribute along with the saccharina to the community diversity.
So in the end, which variable do you think is the most important in driving community diversity, species richness, composition?
Which variable?
Yeah, which environmental factor?
I think that time was the most important variable because we saw that even with the unseeded droppers being bare rope initially, they did develop their own communities by October. So there was some slight differences due to the presence of algal holdfasts versus just growing directly on the rope. But even so, they both had good values for diversity and abundance in October, much higher than was seen for the July samples. So I think time was the more important factor. The longer that you leave the habitat in situ and the longer period organisms have to settle on it, the more diverse it's going to get.
And why? Why time?
Lack of disturbance. If it's replaced every year, it will effectively the community will be set back to square one. And also life history of the organism. Some of the species are the arthropods reproduce over the course of a few months. Some of the longer-lived species need longer to establish. We only saw them in the October samples because they take several months to grow, and if you leave it for another year or 2 years, then other organisms with even longer lifespans might settle in that habitat.
Okay. Can you just go back to the previous slide?
Which one?
The NMDS?
This one?
Yeah, this one. So you see a clear distinction between 3 months and 6 month samples.
Yes.
But you also see that it seems that the variance is increasing with time as well.
Yes, that's right.
How do you explain that?
Can
you? And how would you?
Well, it's explaining the variance over time or between the seeded and unseeded?
No, over time. Well, it seems that the variance is huge after 6 months,
Yes.
both for seeded and unseeded.
Yes. it
While
is.
at 3 months, they're all clustered together,
Yes.
pretty much all similar to each other.
Yes, I'd say it's probably the influence of the algal holdfasts. It creates a different substrate than just the bare rope. So on the unseeded droppers, you have the bivalves, the mussels growing directly on the rope, while with the seeded droppers, you have the rope, and then the holdfasts, and then the mussels, which is a pretty complex three-dimensional structure and favors certain organisms over others. So maybe it has a mix of algal-associated species with bivalve-associated species, while for the unseeded droppers, it's just the bivalve-associated species, which results in the variance between them.
Okay, thank you very much.
Thank you.
Thank you, good Jay. I think we're gonna stop there.
All right, thanks very much.
Thank you. Presentation of this session. So, Lara, whenever you are ready.
So, hello everyone, my name is Lara Laupsha, and today I'm really happy to present you my master thesis project, which is entitled Exploring Cultural Microbial Communities and Prophage Dynamics in the North Atlantic Ocean. So this is a project that has been done with the Danish Center for Halo Research at the Marine Viruses Laboratory at the University of Copenhagen, and under the supervision of Professor Matthias Middelbo and Dr. Sasha Trabi. So before diving more deeply into this subject, I think it's relevant to mention the fact that since the Industrial Revolution, around a quarter of the emitted CO2 have been absorbed by the global ocean, thus leading to ongoing ocean acidification and thereby also affecting the ocean's biogeochemical cycling. So now you're probably wondering, what are the biogeochemical processes that are occurring in our ocean? So CO2 is absorbed at the upper layers of our ocean. Primary producers fix this inorganic carbon through photosynthesis. And so this carbon thus has different fates. In fact, it can either be assimilated back into the food web, it can sink to deeper layers of the ocean, or remain dissolved in the water column. So there are different biogeochemical processes, and I will mention a few without going too deeply into them. So first of all, the biological carbon pump, there's the microbial loop, the microbial carbon pump, and the viral shunt. So in regards to the viral shunt, as you can see in the title, it involves viruses. And here, more precisely, I'm talking about marine phages, so viruses that are capable of infecting and killing prokaryotes. And thereby, they're transforming the particulate organic carbon into dead particulate organic carbon or dissolved organic carbon through cell lysis. And thereby, they're affecting the entire web dynamics and ecosystem processes by influencing the entire flow of carbon. Here in this topic, I'm talking about marine phages, but more precisely about bacteriophages, so phages that can infect bacteria. And so these bacteriophages can undergo two different cycles. On the one hand, you have the lytic cycle, which involves the integration of phage DNA into the bacterial cell, and therefore redirecting the host cell to replication of phage DNA, and eventually leading to cell lysis and the release of viral progeny. On the other hand, you have the lysogenic cycle, which also involves the integration of nucleic acid, but however in the form of a prophage in the genome of the bacteria. And there can be changes in the environment or some environmental stressors that thereby affect the integrity of this prophage and leads to the prophage induction, which leads us again to the lytic cycle. So marine phages can acquire so-called auxiliary metabolic genes, AMGs, from their host. And so these are metabolism-linked homologous genes that are integrated into the viral genome. And so once a virus infects a new bacteria, it can either enhance or change the bacterial metabolic capacities of this bacteria. And thereby, AMGs seem to highlight a dual impact on the biogeochemical cycles. In fact, not only through the viral shunt, but also by altering the prokaryotic metabolic pathways. So now if you take a look back again at these processes, you can see that for most of them, just by looking at the title, you can see that microorganisms play an important role. However, for now, we still know quite little about those biogeochemical processes, not only because of the complex chemicals that are involved in it, but also because we know relatively little about the microbial communities and their functionalities in the entire water column. So this gap of knowledge has tried to be solved in the last decade. You've probably heard about the Terra expeditions, and in fact, they have released the first global ocean catalog that includes up to 33.3 million non-redundant genes. And to do so, they focused on culture-independent metagenomic approaches, which allowed them to cover a large range of genetic diversity in marine bacteria. And it is also important to mention that they increased the sequencing effort of the upper ocean. However, we still know relatively little about the communities in the entire water column. So, in a recent study, it was shown that prokaryotic and viral abundance is actually quite important at the upper layers of the ocean, but there's a drastic decrease along the first 100 to 1,000 meters along the depth gradient, and at the same time that there's an increase of viral to prokaryotic ratio along this depth gradient. So, leading to the aim of this study. So what we know for now is that a lot of culture-independent work has been conducted in the last few years, and in fact it has allowed us to increase the understanding of microbial community composition. However, now we're interested in complementing this metagenomics work with bacterial culture again to explore microbial physiology and taxonomy. What we also know is that viruses are considered as the most abundant biological entity on Earth. However, only 7 to 37% of the marine virus sequences are represented in public databases. So now we're interested in exploring marine bacteriophages and phage-host associations. Last but not least, bacteria and viruses seem in fact to play a role in a crucial role in biogeochemical cycles, and prophages and AMGs show a potential for affecting those biogeochemical cycles. So now we're interested in exploring the role of prophages for host performance and also how the latter is linked to environmental conditions in different marine environments. So leading us to the aim of this study. So here we're exploring the cultivability of marine bacteria that originate from different stations and water depths from the North Atlantic Ocean. And we're also laying groundwork for experimental investigations that analyze the physiology and taxonomy of bacteria. And on the other hand, we're also unraveling the dynamics between bacteriophages and their associated phage across a spectrum of stations and water depths, while we're also screening for potential auxiliary metabolic genes. So I have worked with 16 different water samples that originated from the DPOCA 2023 research groups. And so my samples were from 6 different stations and 4 different depths. And they were all from either the upper layer of the ocean or the North Atlantic deep water. So once I had my samples, I plated them on some marine broth plates. Then I inoculated them and isolated them until being able to pick them to create some freezer stocks. Once I had the freezer stocks, I created some liquid culture which served as a basis for different experiments. On the one hand, I did some DNA extraction for sequencing, and in parallel to this, I also did some phage screening. So for the DNA extraction, I simply used a kit for this, and once we had the DNA content, we sent it off for 16S RNA gene sequencing or whole genome sequencing. And in parallel, I did some phage screening assay, which involved mixing some liquid bacterial culture with some top agar, and then pouring this on some marine broth agar plate. Once that solidified, I was able to pipette some viral concentrates on the latter until seeing a potential inhibition zone that was created. Once there was an inhibition zone, I was able to scrape it off and then reiterate this experiment. So as a total, I obtained 92 different isolated bacteria, and so I isolated them based on differences in color and motility, as you can see right there. And after sending off my samples for 16S RNA gene sequencing, we can see that my bacterial isolates were from 3 different phyla, namely Bacilliota, Bacterodota, and Pseudomonata. And for Pseudomonata, we divided this into the class level simply because this is the most observed phylum that is observed when you do culture dependence analysis. And as you can see, we had bacterial isolates from 17 different genera. Now when we're taking a look at the presence of distinct bacterial phyla in different sampling stations, we can see the omnipresence of Pseudomonata, so that is by looking at gamma protobacteria and alpha protobacteria. And this can also be revealed when looking at the presence of distinct bacterial phyla at different sampling depths, where gamma protobacteria was sampled but also cultured at all depths, and followed by Bacilliota and Bacterodota. So the omnipresence of Pseudomonata can also be seen when looking at the presence of distinct bacterial genera at different sampling stations and depths, whereas Cyprobacter was the most omnipresent bacterial genus, followed by Pseudoalteromonas. And so the omnipresence of Pseudoalteromonas can also be revealed when conducting those bacteriophage screening, where we tested the different viral samples against bacterial isolates. And so depicted here are the formation of inhibition zones after putting some viral samples. So there were 11 successful experiments with successful isolates, and 10 out of these were Pseudoalteromonas, and 1 was Pseudocalibacillus. However, this is a first trial, thus it's not very indicative. This needs to be reiterated. However, it still shows the potential for experimental endeavors in the future. So what we also did was screening for potential auxiliary metabolic genes. For doing this, we used some whole genome sequencing data from 13 different bacterial isolates that were all from different stations and depths. And what we could see was that we obtained some AMGs, so mainly 2 in an epipelagic bacterial isolate, and then 2 from different bathypelagic bacterial isolates. So now, what can we say about this thesis? So in regards to the most prevalent marine bacteria that were obtained through culture-dependent analysis, we can see that Cyclobacter and Pseudo-Ultramonas were very omnipresent, and in fact they were respectively observed at all sample depths and all sample depths except for the deepest. And this also goes with, it has also been a finding in previously conducted studies, as those have found these bacteria to be associated with the microbiota of different marine organisms, but not only, in fact they were also associated with sinking particles. And in regards to Pseudo-Ultramonas, to be associated with chitin-based substrates. Now, in regards to the prospects that we can look at in the future, we can say that Cyclobacter is quite resilient, and it would be interesting to look at the pathways that are involved not only in the initiation of biofilms, as it has been done previously, but into the maintenance of those biofilms, and also to look into the community composition characterization of the bacterial community that is associated with sinking particles, and maybe use Cyclobacter as a model for the study of the latter. And in regards to Pseudo-Ultramonas, based on its preference towards growing on chitin-based substrates, it would be quite interesting to look into the potential presence of host and perfectly encoded functional genes, such as, for example, chitinase in Pseudo-Ultramonas, and see how this actually can potentially affect the biogeochemical cycle in order to get more insights into this. So what we've also found is for AMGs, and what is really relevant is that we have found 2 AMGs in bathypelagic bacteria, which will potentially fill a knowledge gap of deep-sea bacterial host, phage-host associations, which will allow us to get a deeper understanding of how bacteriophages affect bacteria, but also how this is also affecting biogeochemical processes in the water column. So what we've also found is that there is a potential Pseudo-Ultramonas and Pseudo-Ultramonas associated bacteriophages, and that in general there was a broad virus host system range across sampled depths and station, and that Pseudo-Ultramonas could potentially be an ideal host for the establishment of model system to study the relationship between phages and host, and to look also how that affects, again, the different processes, biogeochemical processes in our oceans. Now, lastly, we found a potential Pseudo-Calibacillus associated bacteriophage, and compared to Pseudo-Ultramonas, there was no data that has previously been found in regards to a potential association with a bacteriophage and Pseudo-Calibacillus. So this can potentially lead towards a new finding in this field, which is quite exciting. Yes, so I would like to thank my supervisors for allowing me to conduct this research, and thank you very much for your attention.
Thank you, Lara, for your presentation and perfect use of your allotted time. So.
Thanks.
Yeah, thank you, Lara. It's definitely not my field of expertise. So I do my best, but please apologize if some of my questions don't make sense.
No worries.
It's possible. I've never run a PCR in my life. Okay, so you're using, you're using 2 different approaches, one that is culture independent and one...
No,
you don't
only
use culture independent.
I only use culture independent approach.
Yeah.
I also
How are they complementary and how your culture-dependent approach that you presented here
yes.
can bring something new to the culture-dependent approach
Yeah.
that you presented before?
So in the past, a lot of culture-dependent work has been done, and that was the only approach that has been used for a very long time. And I think it was in the 1980s that they have discovered that when you culture bacteria, you leave out a huge amounts of biodiversity, and this is called the Great Plate anomaly. So when you culture bacteria, you culture around 1% of the biodiversity that is found in the water column. So a lot of metagenomic work has been done since we had the possibility to do so, and that's why, for example, Terra Ocean was so successful at just creating so much data, because it was fast, it was effective, and it was way more precise. However, now that we have this huge amount of data that in regards to that was obtained from metagenomic approaches, we cannot study the physiology of bacteria with just metagenomics data. So that's how my work is complementary to this metagenomics data, is because we now want to study the physiology of them. We want to look at the association with phages, with bacteriophages, and for that you need cultures. But we also want to look into the future. I was not able to do this, but I would love to. Was to look at enzymatic assays and to get a better understanding of the physiology.
So you're going back 40 years backwards?
Yes and no. Yes and no.
That's a joke. Yeah, and what are your thoughts regarding the significance of your results, considering that
Which ones?
of all your results, and especially regarding the identification of phage bacteria systems?
So,
Considering the fact that because you're working with a culture-dependent method, you're probably working on a very tiny proportion of all the systems potentially existing. No?
what do you mean by only the small?
Well, you work with bacteria that
Yeah.
are culturable.
Yeah.
And so do you think that kind of systems and the kind of things you presented are like, does it mean it's much more prevalent in the natural ocean?
Well, it depends. For example,
Yeah.
So what we've seen, for example, okay. So when I did some bacteriophage screening, so as I said, this is still a very initial test. This needs to be reiterated a couple more times in order to obtain some significant results. But for example, we've seen that through culture-dependent approach, Pseudo-Alteromonas was the second most prevalent genera, just by culturing. And what we've seen also when testing against viral samples, that this was again the most prevalent bacterial isolates. And however, when you look at the studies that have been conducted previously, we can see that in fact it is quite prevalent also in the ocean. Why? Because it has the capacity to grow along with the microbiota of shrimp, sponges, corals, sinking particles, chitin-based substrates, which is found in the exoskeleton of arthropods. So I think the reasons why they were capable of growing so well in the lab was also because, in general, in the ocean, they're capable of growing on a large range of different substrates, and yeah.
Okay, answers my question. When we're here, what do you mean by prevalence?
The most omnipresent.
What do you mean by omnipresent? You're talking about abundance, diversity,
I know you
and
would come with
you
this.
see my next question coming.
I knew you would come with this, and I was thinking about you when I was doing my
I'm father.
and it was really confusing because you cannot use the same approach to this
I
kind
know
of study
that's why my
Then-
question becomes silly.
than with metagenomics, and at first I didn't understand it, and then
So yeah, the fact that you find them everywhere when you try to control them,
yes,
does it mean they are the most abundant in the ocean, or does it mean
No.
they're the most
no,
easily controllable?
yes.
I like when you give a choice and when you answer yes.
No, but it is true. You cannot say it because the ocean is for the most part constituted of water.
Yeah.
And here we can
That's
see.
a really interesting.
Show me the graph.
Oh my god. So yeah, I don't I don't know about in the water, but we've seen that it has been efficiently growing on substrates. And there are different substrates in the ocean.
Is it the same?
What?
Is it the same species that grows on shrimps, sponges, corals, and particles?
No.
Actin-based substrate?
Well, around like
Is it? Or is it
no,
not?
no. And for example, no, it's not.
Okay.
No.
Then it's not the same species.
No, it's
Then
not.
it's not prevalent.
No, I'm talking about on a general level.
Ah,
I'm
okay.
talking about the
Okay.
general genus level.
Okay.
Yes.
Valentin has a question?
Oh yeah?
Yes, thank you for your presentation.
You're
I'm wondering if you're interested in like the interaction in this bacterial community. Would it be relevant to screen for like transcriptomic, proteomics, or metabolomics either? If especially with the bacteriophage,
yeah.
I guess it must be like a chemical warfare.
Yeah. Yeah. No, I think
About that.
it would definitely be relevant to do so if there was more time and money, in fact. But yeah, I think what we would really try to focus on right now is to characterize a potential bacteriophage. That would be mainly the first priority, and then to look potentially for the characterization of AMGs that are associated with bacteria, and just the data that I have here is only from bioinformatic results, and you need to test them. So I have found some AMGs. There are some, if you go further, you can find potential functions of those AMGs. So what do they actually confer to the bacteria? So you can make supposition by on the bioinformatic level. However, you need to test that out then in the laboratory. So yeah, am I answering your question?
Oh, yeah.
Okay.
Yeah.
I have questions also, go ahead.
Well, I was a little bit in the same direction with Van Gauthier, but talking about natural selection, like you're identifying here an interesting connections between bacteria, bacteriophage. So my question may be a little broad in general, but how common do you think this association with bacteriophages is in nature, and how could you explain that through a natural selection process?
You mean how bacteriophages are actually conferring a benefit, could potentially confer benefits to bacteria? That depends on the environment. It depends on the potential AMGs that these bacteriophages are conferring to the bacteria that they are infecting. It also, it depends also on the environment, because you might have a lysogenic cycle, you might have your prophage that is sitting there quietly, and then you have a change in the environment and it goes into the lytic cycle, so you have the release of our progeny. Again, it is difficult to answer it very precisely because it is a very broad.
Oh, okay. I appreciate your answer.
Yeah,
Even if it was not one of my questions,
Okay,
but my question was
Sorry.
clear. So I'm not sure if I can make it a little bit clear, but what I had in mind is that we are talking here about fast changing species, like strains. So in order to stay connected together through natural evolution, they have to evolve together. Okay?
You mean about bacteria or bacteriophages?
But both, they have to evolve together so that they can still connect together.
Yeah.
So do you know if you think about that, how they evolve together, or how some can evade that, or coevolution?
Yes. Well, the bacteria has different immune systems. So the first thought is that the bacteria's immune system would fight against the infection of a bacteriophage. However, when that does not occur, you can have a bacteriophage that confers a benefit or not to the bacteria. And when it comes to natural selection, in regards to how bacteriophages are changing and how they're infectable, how the change of viruses are changing their infection capacity. That's your question.
Can be.
I think it is a very advanced question, and first you would need to look into generally the infectability and the host bacteria.
What's the part
bacteria
of the
and
bacteria which can resist infection and which the part does not, and is it positive or not for their fitness? So I know if you have some studies on that, the proportion of the bacteria which are infected, not infected.
No. I think it's also because it's a very more or less a recent field, and so.
Let's jump to
Yes.
Thank you. I again, like my colleagues here, I don't know much about what you've presented us today, but I do know a little bit about community ecology. And you say you want to study physiology, so you need cultures. When you look at the rules for governing the assembly of communities, we basically have three basic filters. There's neutral random processes, there's constraint enforced by the environment, the conditions of life, and then there's the interaction between the species. And if you look at a community and you find species that are present, that are there, well, they're interacting with the environment, they're interacting within the species, they're interacting between the species, and if they maintain themselves and live there and reproduce there, that means in terms of physiology, something is happening with regard to all these constraints. What are your thoughts on, well, you said it yourself, if you culture, you're only taking part of the community, so why is it interesting to look at the physiology in this totally unnatural condition in terms of interactions compared to the reality where there's all these other species present?
That's a good question. Well, we're doing a culture-based approach, and we're only at the premise of those physiological studies. So for now, if we would go further into this, we would just get a broad range or a broad understanding of how the physiology works and how the enzymes that are in the bacteria are working in lab conditions. However, you're right that it would not be representative of how it works in the environment, but it is still a good first step to understand at least the.
I'm not saying don't do it. I mean,
Yes.
that's the best you can do to try to
You have to
approach
start
it.
with that. So.
All right.
But I agree
Right.
that it's not representative. of
Thank
what's
you
in
very
the
much. Do any of you have any more
Yeah,
questions?
maybe
a last one.
final question, and it's difficult to speak microbiology with macrobiologists,
Yeah.
and I
Sorry.
feel sympathetic with you. But I still have a last one.
Go ahead. Quick one.
I was mainly thinking that pretty much every single living organism has viral DNA in every single of its cells. So I was super surprised to see that it was not the case for some of your bacteria where you didn't find prophage DNA.
Yeah.
Is it surprising or is it normal? And
I would have expected more so, but it was not prophage that we're looking for. It was auxiliary metabolic genes. So.
Can you go back to this table? There?
Yes.
The presence of prophage genes. Okay.
Yeah, here it was, so it was not.
Yeah, those ones.
It's
But three.
Yeah, some didn't have prophages. For that,
I'm not
but yeah, but here we mainly looked at auxiliary metabolic genes, so genes that confer a metabolic change.
Okay, I think we're done torturing.
All right, thank you very much, Lara. Thank you And we will now take a break, and for those of you online, we will be back at 11 for the final section.
Okay, here we're back live for the last section, and I hope you will enjoy it. In this session, we will start with Karina talking about climate change and marine food webs, and we'll go with Saish in Honduras, and we'll finish with Ingrid talking about marine pollution and oikopleura. Yeah, you can be ready, yes. And in your jury, you can have Lars Seman from Sorbonne Université, Oxy from Basque Country, and myself, Christophe, from Université Côte d'Azur. Karina, you're ready. Ready to enjoy the moment? Yes? Allez, let's go.
So welcome everyone, my name is Karina Fischer-Bauer and I would like to talk to you about my thesis on the climate change implications on the basis of marine food webs. So I did my thesis at the University of Ghent and my supervisor was Anna Maria Lafayette. So why are food webs and marine food webs important? They illustrate trophic connections among and interactions among taxa. They represent energy and nutrient flows and also describe feeding relationships among species in a community. But more importantly, they provide several ecosystem services, which are in many cases very important to coastal communities. For example, food, culture, or supportive services. Some important ones are carbon sequestration, erosion and flood control, and storm protection. Climate change can reconfigure physical food webs through major structural changes. It can also independently affect or amplify effects of other disruptions, for example, habitat degradation or overexploitation. So we all know how serious impacts climate change can have on marine organisms and ecosystems. Here you can see the drastic increase in ocean heat content in the past 70 years, and it is also projected that the global mean sea surface temperature will increase 2.72 degrees in the next 70 years. This can cause changes in the distribution and abundance of copypods, which is important because they are at the basis of the marine food web, so any disruption at the lower levels will move to the higher trophic levels in the food web. Some indicators for this can be trophic biomarkers such as fatty acids. I think this is an important topic to study because copypods make up 70 to 90% of zooplanktons in pelagic ecosystems, and they are also the most abundant meiofauna in benthic environments together with nematodes. And also there is a very extensive research done on the impact of climate change on planktonic copypods. However, there is not that much research on benthic harpopticoid copypods, which even though they have an important role at the basis of the benthic environment. So the copypods I used for my experiment is Papihelicos imperalis. This was chosen because it is very abundant at the sampling area we went to, and it is also easy to work with because it is a slow-moving copypod. They are primary consumers in marine and estuary segments, and they are also main food source of juvenile fish, for example, flatfish and salmonids, which are very important commercial species. They have wide accumulation capacity to environmental change, and they also require essential fatty acids, EPA and DHA, to survive and reproduce. They obtain these fatty acids from their food, the diatoms they consume, and they also have the ability for fatty acid bioconversion, which means that they can convert short-chain fatty acids into long-chain fatty acids, and this may provide them adaptation to thermal stress. So the aim of my thesis when I started was to look at how climate change would impact the levels of fatty acids in copypods and to take into account their ability for bioconversion, how that would give them a better adaptability to thermal stress. However, we had to change this during my experiment. So the aim of the experiment I will present is to analyze the survival phasing of copypods and growth of diatoms in response to increase in temperature and salinity, and also the presence of oil, which we added as an environmental stressor because oil spills are becoming more and more common. The hypotheses were that the copypod's fitness will decrease, the oil spills will result in increased mortality, and that the high temperatures and high salinity will also negatively affect the diatoms. Materials and methods. So sampling was done at the estuary in the southwestern part of the Netherlands. So after collecting and sieving, we counted the copypods and cleaned them in the lab, and we stored them at the 15-degree climate room at the light regime of 12 hours light and dark until they were used for the experiment. For their food source, we use the diatom nichia, which is constantly cultured in the lab. So the plan was to have a control group, a high salinity, an oil, and high salinity and oil treatments, and to repeat all of these on 15 and 25 degrees, and do all of these treatments with only diatoms, so that I can count the diatoms only and the diatoms in the other treatments, and to compare how much the copy pods have grazed, to compare the number differences. So I prepared high salinity treatment with half of sea salt granules, and we set it to be 38 dpt. The normal salinity level that we used in the lab was 34. And for the oil solution, we used a 10% solution of crude oil and added that to the petri dishes. So for the setup, for the first round, this was only a control round on 15 and 25 degrees. So this was done with the previous aim of doing fatty acid analysis. That is why I used 80 copy pods per petri dish to then have enough of them alive at the end of 10 days to be sent to the analysis. So I used 4 petri dishes on 15 and 4 on 25 degrees, and did the same with diatoms only. These were incubated for 10 days under the light regime of 12 hours light and dark, and I counted them every second to third day, which is when I counted the live and the dead copy pods, and I removed the dead ones and took pictures of the diatoms to then later be able to count them. For the second round, here I did a control group and an oil treatment, because at this time I did not have enough copy pods to do everything in the same time. Here I used multiple plates with 24 wells per plate instead of petri dishes, and I used 1 copy pod per well. So in this time I did a control and oil treatment on 15 and 25 degrees, and did the same with diatoms only. Here I covered the plates with parafilm because the salinity increased a lot in the first round, so here I tried to prevent the increase in salinity, and these were incubated for 7 days under the same light regime, and I also checked them every second to third day, and here I started counting fecal pellets as well, because I was interested if I could see the difference in the fecal pellet numbers, and it was also easy to do with only 1 copy pod per well. And this is also interesting because the copy pods assimilate around 30 to 60% of the food they consume, and the rest they release in the fecal pellets, and after that often they consume the fecal pellets again because they still have nutrition in it. So the third round was when I did all the treatments at the same time. So I did 4 plates on both temperatures. I again covered them with parafilm, again incubated for 7 days and checked every second to third day. However, here you can see these red dots. These were contamination in the high salinity solution, which we couldn't identify. So I repeated the high salinity treatments again, and those came out clear. So I used the numbers from that 4th round and substituted it here to the 3rd, so I can compare everything. So results, these are preliminary results, as I'm still not finished with my thesis, so I'm still working on the results and discussion part. So for the first round, it was very unexpected that the salinity increased a lot. So from the starting 34, on 15 degrees it increased to 30, 40, but on 25 it increased to 43 and 52. So this disrupted our plans a lot because I knew I wanted to do high salinity treatment as well. So I needed to keep the salinity level of the control at the same number. And also you can see here the mortality at 15 and 25 degrees. At 25 the mortality was very high, which can also be caused by the very high salinity. And also at 25 degrees the density of the diatoms was very, very high at the day 10. So that's when we decided to use only 7 days for the experiment because the density of the diatoms was so high it was difficult to count. So these are the results from the second round. Up here you can see the number of fecal pellets per well. You can see that on 25 degrees in general it is higher than on 15 degrees, and also in the oil treatments there are less fecal pellets than in the control group. Here on the day 10 there are no numbers because, as I said, the concentration of diatoms was so high that it was very difficult to spot or to count the fecal pellets. Here you can see the average number of diatoms per 1 square millimeter. So the wells were 1.9 square centimeters, but I decided to use these numbers because this will be much higher. So you can see here that the density at 25 degrees is double than on 15 degrees, and it's double at all the treatments, or at the oil treatment, both at the control, and it's much less on 15 degrees. And also on 15 degrees, the number of deaths was 4 to 5 per plate, so 4 to 5 coffee pods died out of the starting 24. However, here the mortality was also very high on 25, so 17 to 19 coffee pods died out of 24 at the end of the 7th day. So for the third round, is when I did all the treatments. Here above, you can see the average number of fecal sites in the first rows and the number of deaths per well in the second row, so the number of dead coffee pods out of 24. Again, at 25 degrees, the number of dead coffee pods was very, very high, much higher than on 15. And the number of fecal plants was also very high here, but the number of dead coffee pods was also high. And you can see that the arsenic and oil treatment had very low number of fecal plants. Here is the number of diatoms per square millimeter. Here I diluted the diatoms a lot more to have smaller numbers and to not have as dense wells at the end. So that's why the starting numbers are slower, but again you can see how the ending numbers are again double than at 15 degrees. They're double at 25. And also here I had an unexpected result when I counted everything. I expected that the diatoms in these treatments, where there are coffee pods present, that there would be less diatoms than at the plate where there is only diatoms and no coffee pods. And it turned out that there is much more diatoms with coffee pods than without coffee pods. So my plan to calculate their grazing will be very difficult now. So to conclude my experiment, from all the rounds I did, it's the same conclusion that high temperature and rapid increase in salinity caused high mortality in every case. The fecal pellet numbers decreased drastically in high salinity and oil treatments. They were always high at 25 degrees, but coffee pod mortality was also very high at 25 degrees. The diatom density was also high at 25 degrees at all the treatments. It was much higher than at 15. The oil treatment, the oil slowed the spread of the diatoms, so the diatoms formed clusters in the oil, and they also stuck to the coffee pods, so the coffee pods were covered in diatoms, so they were unable to move, which would also cause their death. So I think this is an important topic to study because coffee pods are the primary food source for several marine species. They are important in nutrient cycling and contribute to important ecosystem services. Also the disruption of their population, because they are the basis of the food web, can cause extreme effects without the food web. Also, it is important to understand how they adapt to changing environments and to develop predictive models for the future. And I think it is still important to do more research on their fatty acid bioconversion and the effect of climate change and increasing temperatures on their fatty acid levels, and how that would make them more adaptable to thermal stress. That is all I prepared. Thank you for your attention.
So here we have time for questions, sometime actually, because you haven't used 15 minutes. Lars, you want to start?
Thank you very much for this presentation. Why have you been selecting those two species to answer this question about the relative impact of oil change, salinity change, temperature change? Why these two species?
Divided the species of copepods and the diatoms.
Yeah.
There were two species of copepods that I could choose from this area that we used for sampling, and all my supervisors and people in the lab recommended to use this one because they were moving slower and they were easier to store for a longer time in the lab, so I could do more rounds of experiments with them. And for the diatoms, there was also two options to choose from, and my supervisor recommended this niche.
But in nature, in the natural habitat, will this copepod feed on these diatoms, or?
Yeah, it usually feeds on several, so on a mix of diatoms, not on just this one species, but it was easier to do the experiment just with one.
So you knew that there were interactions?
Yeah.
Okay, and so I didn't get this copy pod is the Atlantic one.
Yeah.
Not at all pelagic. And the diatom is also Atlantic one.
Yeah, yeah.
Okay. How did you choose the level of oil and salinity and temperature?
It was all
Are they
from
realistic compared to what is observed in nature? Or
it
what
was
is
all from
supposed
the
to
papers.
cause global change?
It was all from the papers that I read on this topic and that my supervisor sent me to read on this topic. So the 15 and 25 degree, the 15 is always used as the control temperature.
So 15 degree here is only in winter. So is it regarding what season is it? For example, in the natural habitat.
I think class is asking if this temperature that you saw in the papers, they are reflecting realities in nature in seasons 15
Yes,
and 25.
it's the usual temperature at this part of the Netherlands where we collected them from. So all the papers, most of the papers that I used for source are from this same area of sampling.
And then for the oil concentration,
There was only one paper that we found that we could relate or that we could take as a basis, and they used low oil level and high, and the low was 5 and the high was 10%. So I
let's
chose
say
10%.
let's say, in terms of volume.
Yeah, in the solution there was 10% of the crude oil and 9% of water. Yeah. So I chose 10% because I thought it would bring better, like higher effects.
And
So
when
we
you
could
choose a crude oil, I'm not a specialist, but not oil and the same chemistry. Some oil might be even nocive, I mean bringing death to the poison.
Yeah.
The oil that you choose is the one that is found in the field.
That's true. It was based on my supervisor's recommendation, to be honest, the crude oil, because they were doing experiments with this in the lab, and they said that this is what they use. So
They say it's a good one.
yeah.
Good. At some point you mentioned a temperature of 2 degrees.
2.73, the increase of temperature. The increase of temperature, the predicted increase.
It was in the method I was
This,
no, in the method. No, in your experiment you were using 2 degrees.
the 15 and the 25.
Here also, go back. Repeat on 2. I was a little fast.
So all
Okay.
of these were done at both 15 and 25.
Okay. In the first table that you show, it's unclear what the lines are.
The one separating the numbers.
Yeah,
What
the
are the
number,
different rows?
what are the different
The Petri
rows?
dishes. Because I used 4 Petri dishes on each temperature. So there were 80 copy pods in each Petri dish, and this is how much survived per Petri dish.
Okay,
So
so
from
it's,
80.
it's a Petri dish
Yeah.
number. Okay. I was surprised that some of your numbers have decimals and some don't. For example, the one at the end of the results here, for example.
Yeah, like here. Because the number was so small, like 0.1 or 0.001, so I decided to keep it simpler, but I will include it in the thesis, the whole.
Okay, thank you very much.
Thank you very much for the presentation. Be critic in terms, for instance, your title. Do you agree that climate change, you are looking for climate change implications with your experiment?
Yeah, the result of climate change. Like,
I mean, there are only 2 temperatures in a short time.
yeah.
I don't know, it's because I was expecting something different, because food with climate change with an experiment like this, perhaps is
Too simple, right?
no, too difficult to generate specific conclusions related to the climate change.
To be honest, when we saw that the original plan with the fatty acid analysis was not working, that's when we tried to find a more simpler approach. That's why we used the multiple plates to use less copy pods, and also we shortened the duration from 10 to 7 days, because also the diatom density was so high at 10 days that it was very difficult for me to count, because the screen was just full with diatoms. But yes, you're right, it's difficult to draw conclusions from 7 days only.
For me, just as a recommendation, because I mean, if you put temperature, different temperatures, the response to different temperatures, the effect of different temperatures, and then you justify, because of course it's important to know what is temperature doing in a climate change environment. Okay, I mean, it's more accurate, perhaps. So sometimes it's we try to say too much, and but I don't know if it's the right way to do. And also you plan the objectives and then the hypotheses. And even more, I would say that there are not hypotheses when you are planning, because the hypothesis is something that is based on the previous knowledge, that you make a question, but it's not a question. I mean, it should be something. And then you plan the objectives just to prove or not that hypothesis, just to keep in mind, because sometimes we miss the idea of academically how should be, scientifically how should be, and I think it's important also, because, can you put the slide? You have the objectives, and then even I would say that there are no hypotheses. Copper business decreases. It's not an hypothesis. It's not the.
It's more expectation.
Yeah. Okay. This is happened to all of us. I mean, there's no. It's more just to be aware of different things. How you design the? I mean, I didn't get it. You put the copper pods directly to temperatures, to different temperatures from. I mean, is a heat shock happening, or do you increase the temperature along the time?
Because they were stored at 15 degrees, and the ones that on 25, they were immediately just put at 25. It was not, there was no gradual increase.
This is more like a heat shock.
Yeah.
And sometimes it's more difficult even to relate with the climate change that is supposedly gradually happening, depending on the placement, because it could be events of heat shock as well. But this is important, and also it's important for the salinity, because of course the water is evaporating.
Yeah, to increase it gradually, right?
How you could change the design just to avoid that?
Maybe to increase the temperature and strength every second to third or fourth day.
But to avoid the, I mean, once you increase the temperature, to avoid the evaporation, or maybe another type of design, tanks, whatever?
I covered the plates with parafilm, so the water couldn't evaporate.
Okay, yes.
Because after the first round, when the salinity increased so much, then I took a few weeks when there were no copper pods to do other experiments, and then I just tried the water on different temperatures with different plates and parafilm and put them in boxes and different methods to see how to keep the salinity at the same level.
Yeah, okay. Okay, thank you.
So maybe following on that, how do you distinguish the effect of temperature and salinity and everything on diatoms and copper pods? Because you were looking at do you have a possibility to distinguish the effect of on copper pods and on diatoms, or do you see only the result combined? So, in other words, are diatoms pre-acclimated to temperature and salinity?
No, it was also the same with copper pods. We put them immediately at 25 degrees with the high salinity. Okay, so it was no acclimation.
So how could you be sure that it's not the food of the copypod that could be impacted as a reason of the death, as a reason of the copypod death? Oh,
This is
sorry.
a good question.
Your internship is ongoing, so we're just giving just discussion. I'm curious also how you were selecting and manipulating the copypods to put them in the wells, because like, it can be super hard. One copypod per well.
Yeah.
So with a tiny.
It was with a tiny glass pipette for hours. yeah.
And you had a way to selecting them, like you were selecting the most giant, the fastest ones, the less fast ones, or it was random.
It was random. Just they had to be alive and moving, and then we selected.
And you were making sure they were still alive after the process, I guess.
After the selection.
Once they were in the well.
Yeah, they were alive. Yeah.
I have a few more questions, but last.
I was wondering what kind of statistics would you use with your data? Because in the next months you are going to continue to work on it.
Yes, that's what I'm currently the part where I'm stuck at because I'm constantly running at errors. So now I'm talking to my supervisor to try to figure the result part out.
So here you were using 25 degrees for copypods. Some of them were dying. Do you have an idea of in the literature of the LC50 or those lethal temperature of half of them in general? So at what temperature would you expect them to die from the literature?
Mm-hmm.
Not really.
I don't know, I'm sorry.
And also for maybe my last one, if you have others. My last one will be the fecal pellets. When you talk about fecal pellets, can you go on a slide where we have fecal pellet numbers?
There.
The first. Okay, these numbers correspond to what is a unit actually? It's like
it's a number of fecal pellets. So I counted the fecal pellets in each of the wells.
Okay.
And then these are the average numbers because there's 24 per plate. So
for both alive and dead copypods.
Yeah.
Okay. Maybe it could be interesting to refer only to the fecal pellets for the still alive copypods. To see if the still alive copypods are in as many pools as before. Because if you combine both, so you're not maybe looking at the metabolism of the still alive copypods, but
yeah, for example here on day 7, I didn't count the fecal pellet of the copypods that were dead.
so it's only the still alive copypods.
Yeah.
Okay, good.
So coming back to my unit here, the fecal pellets are it's an average, therefore you can have decimal values, but the number of copypods dead, it's not an average. It's
No,
the total number.
it's an exact number of dead copypods, yeah.
And what did you do? I think for one result you choose to make an average, for the other result you choose to display the total number.
Because there were 24 wells in one plate, so in each well there was a different number of fecal pellets. So that's why I combined the number of fecal pellets for one plate, which is one treatment, but this is the number of deaths for the whole. So from the high salinity, from 25 copypods, 19 died.
Great. So your work is ongoing. So questions were also to help you maybe direct your work for the future weeks and months. So good luck for the continuation of your internship and congratulations again. We'll move on with Saish. Just a second so that everybody comes back. Now, are you ready, guys? Yes. Allez, you have the floor.
Okay, thanks you all. Thanks everyone watching online. I'm excited to tell you about my research that I got to do in Honduras and Telabé. We were using empirical indicators to describe this multi-species data-poor fishery. So I'm going to tell you a bit of background about the work and why we chose our approach, and then I'll tell you some of our methods and results. We did a lot of work. I'm not going to get to tell you all of it, but I'll give you the main results, and please feel free to ask me questions. So fisheries generally classify into industrial and artisanal/small-scale fisheries. This is kind of standard. I'm not giving a rigid definition for SSFs here because they tend to go on this spectrum with various characteristics that are generally near shore or coastal, multi-species or multi-year, very important for our fishery, and subsistence-oriented. So catch is mainly for consumption, not exclusively, but mainly is generally how we understand it. Despite this, they account for half of all global fishing and most consumptive-oriented catch. So they're really, really important, and they've been quite understudied because traditional assessment and modeling techniques are generally developed for industrial fisheries. That's where their basis was. So they tend to apply quite poorly to SSFs, and that's also what we found in our research, that the needs for data and expertise are really high, and so that's very challenging in resource-limited contexts. And they also can't really model complex multi-species or multi-gear interactions. They tend to focus on one species, and when you try to account for multiple gears, you have to do separations into fleets, which gets quite complicated. So empirical indicators are an alternative. They are a simpler approach where you, instead of doing a complex population model, try and highlight trends and patterns on simple fishery-dependent data. What I mean by that is just data associated with the catch and the landing. So caught numbers, weight, sizes, fishing effort, among others. They are descriptive, so crucially they don't estimate things like the size of the fish population or the optimal level of fishing effort. That's not the point. The point is instead to describe the fishing pattern, which is how any amount of fishing effort will be distributed across the species or the sizes that are targeted. This is really important for multi-species fisheries, usually SSFs, because it's been used quite extensively for assessing the ecosystem impacts of fishing. A lot of this work has gone under this framework of balanced harvest. Again, I wish I could tell you more about it. It's quite interesting. But the general idea is that you distribute a moderate mortality of fishing across a really wide range of species and sizes. This is in contrast to traditional management, which tends to advocate that you catch really large fish only. And instead, you exclude the smaller fish so that they grow to a recruitable or an optimal length, and then they can recruit. There's a bit of a trade-off in these two approaches. The idea with this one is that the ecosystem structure of the system is preserved, whereas when you do single species, large size fishing, you can induce fishery-dependent selection against large sizes, which makes them smaller and smaller over time. So with that in mind, our goal was to develop an analytical framework for describing multi-species SSFs in the Caribbean, focused on the fishing pattern, and Telabé was our case study. So specific to Telabé, our sub-objectives were to describe the fishing pattern for this species, the lane snapper, which was commercially quite important in the region and has been previously studied. So we wanted to link that assessment. We also then wanted to look at how this species dynamics interacts with the pattern for the multi-species fishery, and then how both of those patterns have changed with the introduction of fisheries co-management, which happened in 2018. I'll tell you a bit more about that. So what did we do? For one, this is Telabé. This is the main site. It's in the north coast of Honduras, and there are 6 communities mainly involved. It really epitomizes the challenges of SSFs everywhere. It's got limited resources and infrastructure. Enforcement capacity for command and control management is quite poor. It's subsistence-oriented, and mainly it is multi-gear, multi-species. So about 50 species are caught in this small fishery, and they use about 12 gears, 4 primarily though that I'll be focusing on. In 2018, the Coral Reef Alliance, my partner organization, and the fishers in the community collaborated on this co-management plan. It interacts with a lot of other jurisdictions and laws that are kind of represented here, but the main things to know are that it introduced these size restrictions on the lane snapper in this framework of exploiting fish at large sizes. That was the original intention. So a minimum size of 23 centimeters, a minimum mesh size of 3 inches for gill nets, these fish recovery zones that are marked here in green, where gill net fishing is not allowed, but crucially handline fishing is permitted everywhere in the region. The data that I used was also a community-based initiative. Coral Reef Alliance has worked with community scientists to track landings once a week since 2015 in all 6 communities. It's a really, really cool effort, and they capture a lot of fishery-dependent data, But there's quite a bit of variation in the sampling effort between communities and between years. The COVID-19 pandemic had a big impact on this as well. So we couldn't really do a time series assessment per year. Instead, what we had to do is group the data before and after co-management so we could do a descriptive analysis. So I'm going to say before and after quite a lot, just so you know that is before and after co-management. For the length snapper, we had two objectives. The first was describe the selection pattern through a length frequency distribution. It's a standard tool, but overlaid onto that standard tool, we talked about these catch sustainability indicators, which have been used in the past to assess this fishery. There's three targets there. They look at the length of maturity, the optimal length, and the length of megaspawners. And the general idea is that a sustainable fishery is above or at the length of maturity within an optimal length and excludes megaspawners. So we don't catch fish that are too large. These are all estimated based on empirical equations that have also been developed by the same researcher, Rainer Frost. Next, we wanted to look at the impact of the gill net selection pattern on theoretical yield. I really emphasize theoretical because this model, the Thompson and Bell yield per recruit, is usually used as traditional stock assessment, but we don't use that in that way in this context because there's a number of assumptions that are violated that we can't rely on. But it's still a really suitable model for looking at the impact of a mesh selectivity regime change. So that's what we do with this model here. When it came to multi-species, we once again used the length frequency distributions, but now we looked at all the species all together, and we overlaid the trophic level distributions on that. And I'll tell you a bit more about that when we get there. The next goal under the scope of balanced harvest was to look at the pattern of harvest by size and by species. For size, there's this complex indicator with this fancy name, log normalized biomass spectrum. But the idea is that there has been a considerable amount of research which has shown that marine biomass is distributed very stably across log-sized bins of size. So the idea is that when we compare the distribution by size in the catch to what we know is a stable ideal distribution in the ecosystem, how far it deviates can tell us how balanced is the fishery and how unbalanced it is. With the pattern of harvest by species, this was just looking at how much was landed per species. We really only use the top 10 because they account for 85% of the catch, so the trends are a bit clearer. What did we find? We find a lot. Let's start with the length snapper. So the first thing I want to draw your attention to here is the length frequency distributions. These are lengths on the x-axis, and this is the count of frequencies on the y-axis, and this is split by the 3 years used for this fishery. The first thing you see is that selection patterns shifts to larger sizes in the after period. This is good because this is compliant with the regulations that were put in place. These indicator lines are 1, the minimum mesh introduced legal size, 23 centimeters. 2 is the length of maturity, and 3 is the megaspawner length. So ideally, we want to be above the minimum size at the length of maturity and excluding megaspawners. And that's what we find, which is really good. This pattern, again, based on the indicators, is considered improved. When we modeled that with the Thompson and Bell yield per recruit model, there was a lot of work that went into making this. So I'm not going to get into tell you all of it, but what I want you to look at is this mark, the F0.1. That is an understood indicator of a sustainable fishing effort. And when the X, which is really tiny here, unfortunately, is above it, it's considered not optimal exploitation. So what we're actually finding is that when we compare selection pattern before and after, that X moves onto the F0.1. So it's more conservative relative to that target, but also the modeled yield that we get, as you can see based on the y-axis, is higher. So essentially, theoretically, if the fishery was in a stable state, all the assumptions were good, we're actually having more fish with less effort with the new selection pattern. So this is a good sign for the introduced gill net regulations that were added by the co-management regime. With the multi-species fishery, again, there's a lot going on, but the two things I want you to take note of is that there is this dominance of the 3-inch gill net in the after period because both of these gears decline, and this one is phased out because of the new regulations. The targeted trophic range is pretty high. It's above 3.6, which generally refers to mid-level carnivores, and herbivorous fish are pretty much excluded. So from the perspective of balanced harvest, you could say that this is just inherently unbalanced because we're only fishing a narrow segment of the marine ecosystem. There's so much that is being left out. This is where I think context-specific factors are really, really important because you can never really exploit the entire marine ecosystem. We don't need phytoplankton, we don't need zooplankton. Really, at the end of the day, we're targeting fish, which is a narrow segment of the marine community. And in the Caribbean especially, herbivorous fish are really, really important and declining because the sea urchin has been lost in a number of places. And so trying to advocate for exploiting more herbivores based on just this would be pretty irresponsible. So this is how we're interpreting our results, is that even though the traffic range is restricted, that's not necessarily a bad thing for this specific context. For the balance of harvest by size, the general way this indicator works is that you have the density of biomass in these log-sized bins. And on the descending part of that density curve, we fit this linear regression. And in an ideal slope observed in natural ecosystems, that slope should be -1. It's not -1 for both. It's quite high in both periods, but much higher before. Kind of improves in the after period with a reduction in the density capture of small sizes and an increase in the density capture of large sizes. I'll say that this is not a significant difference, but the p-value we found when comparing that is small. And more interestingly, this is not what we expected. What generally happens when we put in place restrictions to larger sized fish is that this part of the curve actually steepens because we're harvesting it more. And so you'd expect that there is a loss of that biomass at the higher density range. So usually the trend is to steepen. Instead, what we're seeing is the opposite. There's an increase in the large sizes and a decrease in the capture of small sizes. So we were kind of interested, like, what's going on? It's worth digging into this. And the first indication of what might have been happening was looking at the species balance. So firstly, overall, there was a reduction in caught weight before and after by quite a bit. But the relative reduction for the lane snapper, which is the commercially important one, was the biggest change. And in fact, almost all of the other species actually increased fairly evenly in how much they contributed to the catch. So this one, which was previously quite dominant, has now shifted to a more balanced pattern, you can say. And this we found to be quite interesting. And it has driven our hypothesis for why we find this evidence for improving balance in the harvest of fish, despite there being a very single species management regime in mind. And our hypothesis is that the regulations that were put in place retain the flexibility for these fishers to change their choice of gear, species, location. So this graph shows length and the catch per unit effort per length class for the lane snapper. I'm showing you this because it first highlights that the catch per unit effort in the before period for the lane snapper was really big. That's what we expected. It was exploited across a very big size range, but the CPUE is pretty stable for all of that size range. And this is a standard characteristic for a heavily exploited fishery. When there is a limitation or truncation of that size range based on regulations, if there was no flexibility in that fishery, what you'd expect is that CPUE will decline because fishers now have to take all that effort and fish a very specific part of the ecosystem. But that's not what we see. Instead, the CPUE in the after period per length class stays pretty much the same. These are confidence intervals, so they're actually crossing each other quite a bit. And this is despite an overall reduction in the caught weight for this species. So we'd interpret this as saying the fishers are taking that effort that was going to the lane snapper and now saying it's not worth it anymore. We're not catching the same catch per unit effort. We're going to now catch other species. And so the unexpected consequence of that is that there's a rebalancing across the ecosystem in terms of species and sizes. And that's surprising because these management measures tend to conflict and the literature generally finds that they don't align with each other. So we were excited to see that there might be this potential for a synergy. And I think that's what I want you to highlight as the main take-home message. So this was the first time we characterized, or anyone has characterized a multi-species fishing pattern in Dela and in Honduras. And the main things we found were that the regulations were good for the lane snapper, which is what we wanted. But they were also maybe improving the balance of fishing pressure across these species and sizes by reducing the incentive to exploit this one species so intensively and instead increase the incentive to balance that pressure across the species of the size community. This is really cool because this potential for synergy between single and multi-species management could be really important for small-scale fisheries. They don't have the resources for command and control management to put in place total allowable catches and transferable quotas and impose specific sizes on like 50 species. When I got to Coral, that was the intention. They wanted to find a way to put in place minimum length sizes for all of these species. And we were hopeful that this research has kind of shifted the conversation a bit to try and think about it in a different way. Of course, our results are only as good as the data quality. There were challenges with data quality because of the way it was gathered. But even if we say the results are limited and only demonstrative, what they do demonstrate is compliance with the regulations, and that compliance with the regulations could mean good things for this fishery. So we're intrigued that this research could be shown back to fishers and saying there is incentive for us to actually follow the regulations that you yourselves have put in place, and to support the fact that the co-management regime, which was only informed by traditional ecological knowledge, actually had a really good understanding of the dynamics of the fishery. And the regulations that came from that can be supported by our quantitative research, not necessarily erased by it. Just to end, these are only some of the people that have been so instrumental in helping me in this work. All the code for this research is hosted if you would like to see it. And to conclude the work, we're trying to build this web application, which will continue to track these indicators over time. So the idea is that the fishers don't need me anymore. Once this application is in place, it's going to plug into their data monitoring process, and they can continue to track these indicators and see how the fishery evolves over time. This is expected to be done by September. This is the draft. It looks kind of ugly. But that's all I have for you today. Thanks so much.
Thank you, Sej, and the crowd. So let's start with you.
Yeah, okay.
Okay.
Thank you for the presentation. I think we can feel that you are quite happy with it and you enjoy. That was the point. What was the main problem?
Oh my God. There was a lot of challenges. I think I'm happy with the results that we found ultimately, but this was not the intention. The way the project here was not the intention going in. The original goal was to use those traditional models that I was telling you about, yield per recruit and surplus production. And the challenge was beating my head against them for months and finding that they just don't apply. There's so many assumptions baked into the applications of those models that make them fairly inappropriate for using in this context. And it was really wrangling through the weeds of the assumptions and the math on those to finally decide that this is not the framework that we should be using. And instead transitioning to something that was mathematically simpler, but actually told us much more about the fishery. And in my opinion, continue to give the agency for decision-making to the fishers, rather than having a very complex, expert-driven model to try and determine how the fishery should be managed. So that was hard, because that happened in like April and May, and I had to reinvent the project quite late. But I was satisfied with how they did.
Don't say that the fishermen don't need you anymore, because it's not, I mean, it's better
Yeah.
to continue with the relationship.
Definitely.
Even if it's online.
Yeah, of course. But they don't depend on me, which is crucial.
That's okay, and that related with the data quality. How do you guarantee the data quality? I mean, how much they lie?
That's a good question. How much they lie on the database?
I'm fishing. I go fishing.
Yeah.
And I know that all the fishermen lie.
Yeah.
All of us. Same. Everywhere.
Yeah, I think it's a really good point. I feel like there's reason for hope, because the monitoring program was collaboratively put in place. Like the fishers agreed to do it and have volunteers. Like the community scientists come from each of the communities and they receive training from Coral and then stay there and continue to track the catches. The other positive indication is that in the data we do see illegal catches. So once the minimum mesh size was put in place, we are still finding reports of things below that size. I think that's a good thing. It shouldn't be penalized, especially because with the new framework under a balanced harvest approach, it's not really important to have a hard cutoff for size. It's more important that the size range is tending towards a certain area. That said, that doesn't mean that's not happening. Surely it's happening. But I think what was interesting is that even if we assume these results are demonstrative, they show that there is compliance, like I was saying. So if we see that with compliance, there are good benefits that are coming from it, we can go back to the fishers and say, it's worth reporting, it's worth being part of this process. And Coral has been pretty clear that this data is not shared with anyone. It doesn't get shared with government. I couldn't share it with IMBRC. So it's very well preserved. So I think there's trust in that relationship.
Completely agree. It should be based on trust.
Yeah.
And for instance, in the Basque Country, the ASCII organization that is very involved in fisheries and so on, I think they obtain somehow trust with the fishermen. And it's not the same situation, of course, it's not the,
right.
But I can feel that the fishermen, they trust on what they say, and this is wow.
Yeah, yeah, it was cool. And I think it is why I feel like the regulations have so coincidentally produced these good ecosystem results, is because they weren't imposed top down by managers like me who would be trained in like very specific techniques, but rather based on their understanding of the system. And there were conflicts. There's a report that Coral has produced on the process for producing the co-management, and they talk about conflicts between the communities, between which regulations were, more contested or not. And then what was finally decided was just a consensus amongst all of them, which was also nice to see.
One question, just from the formal aspect. They have 2 months test.
Yes.
It was difficult to follow something because for the audience as well. I mean, when you are presenting something and you put text, we should be able to read in the time you give to us.
Yeah.
If
And I was
not,
flying through it also.
yeah, because you are living on the time and it happens to all of us, but it's important also to be conscious of that.
Okay, I appreciate that.
Nothing more. Thank you.
Yes, thank you for this very lively presentation. Maybe my first question will be about the normalized biomass distribution that you observed before and after. So when you say before, it's how many years, and after it's how many years?
2015 to 2018 was the before period.
So 3 years.
4 years, including 2015. 16, 17, 18. And then after is 2019, 21, 20, 21, 22.
Okay, and those are average biomass?
That's a great question. Let me explain this indicator. The way this works is that you take all the catches from the period, all the species, you group them into bins of weight that are log size, so they get bigger over time. And then biomass density first calculates the total biomass in each bin.
So when you say weight, actually you weight
The fish, yeah, all of them are weighted. So total biomass in each bin, and then you normalize that by the width of the bin, which is based on the log scale, and that's why it's biomass density, so it's per width. And the idea is we only use the descending part of the curve because that's the part assumed to be under full selection. If the whole fishery was being selected for, this would be a straight line all the way through. And I'll note that this is probably also an underestimation because based on our results, the selection at the largest sizes is probably quite low. It's actually being, in our opinion, excluded. So this is probably a bit higher if it were under full selection. So these are probably a little more pessimistic than they might actually be, but yeah, they're indicators.
To be sure, you have only one line, but that line is an average of all the catch during those multi-year studies.
This line, the regression line?
No, not the line, the curve.
The curve.
The data point.
The curve, honestly, the curve probably doesn't matter. It's the points that really matter. The curve is illustrative because it's the standard.
Each
weight.
point is an average.
Each point is the biomass density.
But is it an average or not?
Total, not an average. The total biomass density. For if so, if this bin
4 years.
Across 4 years, all of it. Yeah. The reason it works as a total rather than an average is that what we're interested in is the distribution across sizes, and we had to make the assumption when we did the grouping of the data that the periods reflect overall similar states. So if the fishing pattern in the whole period is the same, we can take the biomass density altogether rather than averaging. If we did the average, which I did try, the pattern, it's not very stable. So in order to construct this graph, I had to test many different bin widths to see that the pattern is actually stable. If we average, then the pattern stability starts to deteriorate, so we can't trust that indicator as much anymore.
Okay. And then do you use the slope of this navigation?
Yeah, so the slope is estimated by a regression line, and this is based on this paper and a lot of other research that this researcher has done. And the slope of that regression line is this b value, which unfortunately is quite small. It's minus 1.96, minus 1.52, and the ideal slope in a balanced community is minus 1. So it's steeper. Does that answer your question?
Yeah, no, but I know this NBSS and the theory behind it.
Uh-huh.
I know it's very sensitive to the size range that you are looking at.
The-
It's too narrow compared to the whole system size range. You might have some local variability.
Yeah.
And therefore, it's very important to look at the sensitivity of your p-value, the slope, given making average or making some. Maybe you should test and explain to the public the robustness of your finding, because maybe it's only a matter of choice. Because you said that you tried many ways of building the size.
Yeah.
And they were not successful, and you decided to present us the one which is successful.
No. Let me rephrase. I tried many ways when I took the average. When we did the total, which was after review of the literature of it not working, the bin width did not matter as much. Actually, the reason I chose this representation is because it's not statistically significant. There were occasionally ones that were, but I didn't want to like p-hack and only show you the statistically significant one. But what we tested was a number of different bin widths on the log scale. So it was 1 to 2 on 0.5 increments of partial bin widths. And we tested all of them, and we looked at the variability and found the trend to be quite stable. But this is 1.5, right in the middle. In my opinion, just visually, it represents the pattern the clearest, mainly because this peak is evident, and this peak is kind of important because this
How do you
is...
interpret the peak?
It's lane snapper. It's when the lane snapper regulations were introduced and the gillnet regulations were introduced, the selection pattern shifted to a larger size. And so then you get this peak right here, but also this slight increase towards a little bit of a larger size, because the gillnets, you've done fisheries, I imagine, you can know this, is that they have this dome-shaped selection curve. Sorry, I'm not going to have time to see this, but this was using the select model to fit a retention probability. And in both cases, whether you have 2 gillnets in the system or 1 gillnet in the system, it's always this dome-shaped selection curve. And so when it shifts to a larger size, you'll have a peak over here or over here, but the selection here will still be a bit lower as it starts. Does that make sense?
Yeah.
Maybe the discussion can continue with sort of following coffee.
Yeah, I'm sorry.
No, but it's very important
No.
that
Yeah.
you look at those details which you did, so
Yeah.
that's a good point of
It seems to.
the study. But, well, no.
No, get hydrated. No, you've done a very nice presentation. I particularly appreciate nice scientific stories. Like it's what you say is accurate, but we can see that yes, you're really aware of contextualizing all of what you have. Just to say at some point, we don't need phytoplanktons. I don't like that. But,
We don't eat phytoplankton.
we don't eat phytoplankton.
We need phytoplankton.
Okay, sorry. So you've got a lot of information. You started talking a bit about that, but how do you come back to the fishermen now and explain that? How do you apply your findings to the community?
That's a great question. Right now,
Thank you, Sage.
doing great. This is mainly oriented towards managers. It's still a technical product. I mean, we're making it available on a phone. So if fishers are interested, they can look at it. It's nice to see pretty graphs. But it's mainly for regional managers to be able to control, or at least inform, how we interpret regulations as we go forward. Because from what I understand, the co-management regime right now is a trial, and hopefully it will be an adaptive process. And my hope with this is that these results should try and center the adaptivity rather than trying to say there is one single way to do this well. So when we use something like this to continue to track how these dynamics proceed over time.
Yeah. But who are you going to address?
Who specifically?
Yeah, the fishermen. There is like a kind of a box of the fishermen of the community.
There's some communities have like a fishing leader,
Yeah.
and they're the ones that are involved. I think some of them have fisheries associations that also have elected leadership that are involved. But I'm mainly interacting with Coral and my supervisor Antonella, and Antonella is very well connected to all of these networks.
With the community.
Yeah, so she's the one that's going to be working with the dissemination, not really me.
Really quickly, I like to have a look in one detail each time. So, what type of corn producers were caught? I think in one of the graphs, the graph with this one. What's the green part?
It was 3 fish species.
Fish.
Fish. It was all fish.
It's 2. Okay, I thought it was 1.
Yeah. Still two, and they were like herbivore, omnivore.
Okay.
Yeah, very
Wow.
few. And I looked into it as well, and the number of catches for them were also
Right.
12.
So another tricky question. You said so. The idea is to try to avoid catching the big fish.
Herbivorous fish.
No, the big ones, the mega spawners.
The biggest one.
How do they do that, the fishermen or the fisheries management?
It's another good question. In this fishery, it's
Thank you again.
in this fishery, it's just a consequence of the selection of the gears that they use. The gears, at least the gillnet, is the most impactful on the larger sizes, and the gillnets have the fortunate circumstance of being with this dome shape selection. So they peak at a certain size, and then they don't really catch anything bigger. So there's no gillnets bigger than this right now.
Okay.
For now, based on this, we would not advocate
So
for
the
having
fish
gillnets.
cannot enter into the net when it's
Yes, and when they're too big, they kind of just bounce off.
okay.
So this is the idea why they exclude small and big. But the hangline is much less selective, actually. And it's, at least you can see this kind of here.
Yeah.
They catch large sizes quite a bit.
Okay. We have to
Yeah,
finish
go ahead.
that. Thank you for your answers. And thank you, Saish, again. Congratulations. And here we go for the last presentation. Well, for the last presentation.
Okay.
It's always nice to finish, to
Yes.
conclude a long set of presentations, so enjoy it.
Yes. Thank you all for being here today. I am Ingrid, and I am really happy to share with you my project that I've been working on these months. It is about understanding the effects of underwater noise pollution in the behavior of this organism, Oikoplera dioica. This research was conducted in Bergen, Norway, and it's part of the Deutero Noise project, GPI Oceans project. So marine organisms, as you know, rely on sounds for basic vital functions, such as reproduction, navigation, and communication. And therefore, they have evolved a variety of receptors for sound detection. The soundscape of the marine environment is, of course, composed by the noise that marine organisms naturally produce, and also of noise of other events like wind, waves, or rain, natural events, and of course the noise that human activities produce, such as maritime traffic. So this noise from human activities overlaps with the hearing capabilities of the marine organisms, and due to the increase of these activities, we want to understand what the effects of underwater noise pollution are in these organisms. So there are many studies trying to understand these effects, mainly focused in big organisms like marine mammals or fishes, and they have found physiological effects or behavioral changes, and also effects in embryos or larvae. However, in this group of organisms, marine invertebrate lithostomes, the effects of noise are not really understood or studied. So I will be focusing today in this little organism called Oikoplera dioica. This animal is a pelagic tunicate, and it has a very important ecological role. This is a component of the zooplankton communities, and it also has an important position in the trophic chain. It has also a very adaptable life cycle that depends mainly in temperature. And it has a really interesting characteristic that is the production of a house. So here you see this is the head of the animal, this is the tail that they use to swim, and all this structure around it is the house. It has a filter, and so this organism is a filter feeder. And they rely mainly completely in the house to obtain the food that they need. It is also very well characterized in terms of swimming behavior or culturing conditions. So this makes this organism a very suitable model for this kind of studies. Here I have 3 videos that we collected in the lab, and here the presence or the absence of the house is mainly related with the swimming behavior of this animal. So in the first video, we can observe the in-house behavior, that even though we don't see the house because it is completely transparent, we know that the animal is in the house because of the rhythmic movement of the tail. If we compare with the second video, the house living behavior, you can see that the tail starts doing more deliberate movements, and they swim faster and all around the arena. And when the animal starts building a new house, they do this movement, and then this little dance that they use to inflate the house, and therefore start eating again. they do this every 4 hours, more or less. So for this study, I had 4 main aims, starting with setting up tools to analyze the behavior of this animal. And for this, we propose to use machine learning, which is a novel approach that has not been done in this organism or for noise, underwater noise pollution studies. And we want to understand what the noise effects are in the swimming behavior of this organism, so how it swims, but also how the house pumping behavior would be affected by noise. And lastly, we want to know what the threshold of noise effect is in this organism. So starting with the first aim, a key part of this study was to actually develop these tools. As I already said, it has not been done in this animal. So luckily enough, we had already developed technology in the lab that is basically a camera that has a magnification lens, and therefore it allows us to record some videos of this organism. This animal is pretty small, like 1 millimeter in size. So with this technology, we could record animals, the animals, and use these videos to train a model using a software that I will explain later on. So here we have one video example, and I already mentioned that the house is completely transparent, but we want to analyze the house pumping behavior, so that was the first challenge that we had to overcome, how to actually record the house, because yeah, we cannot see it, right? So we were experimenting a little bit with some dyes and didn't succeed, but we discovered that giving them a shot of cyanobacteria, Thesis species in Ecococcus, we could actually stain the house, and we could even see when the animal is eating the inner structure of the filters and also the pumping activity. So we decided to, during our experiments, always give them some cyanobacteria that is something that they already eat. So I recorded some videos first, no noise involved here, but some videos to train the model that will allow us to analyze the behavior of this organism, comparing normal behavior with when we expose them to noise. I used this software, Deep Black Cut, and I started by doing this schematic that shows some points that I labeled through many videos and many frames of different animals in different positions and without a house, with a house. Yeah, so the software can recognize the animal no matter how it is behaving. So after that, I obtained this model that can recognize as well the head of the animal and the tail. As you see, the color dots are the same here in the schematic. So we know that the model is recognizing these body parts. But for the house, it was really, really complicated. So we decided to take a different approach and to analyze animals that are inside the house and their pumping behavior. We created chemographs, which we used Fiji software, where we introduced the videos the animals were in the house, and we obtained this, basically the image that tells us how the house is moving. And then we analyze these images with Chimograph Direct, another software, that allow us to extract the data that we were interested on. So for our experiments now involving noise, we had this tank with an underwater speaker, a glass container that had the animals inside, and this paddle just to move the water and avoid the animals to sink and die. We did 3 main phases in our experiments, and it was starting with an acclimation phase of 1 hour, which basically we put the animals in the beaker for 1 hour, no noise, and then we took them back to record them under the animal tracker that I showed you before. So we took them with a pipette, we put them in an agarose arena that we prepared in the lab, and we gave them the shot of cyanobacteria. And then we exposed the animals to noise, putting them back in the beaker, turning on noise for 1 hour, and then record them again for 15 minutes. And we repeated this with the recovery phase, of course no noise. We did this in triplicates for 3 different noise levels, 11, 20, 10, 15, and 20, with a background noise of 138 decibels. The sound profile that we used to expose the animals was a continuous ping noise. So now, going to the effects of noise in the swimming behavior of this animal, I used 3 main metrics to measure the effects of noise. I started with using frequency, which refers to the tail beatings per minute that the animal do, velocity, which is the speed and distance at which the tail moves, and amplitude, which is the displacement of the tail. And for this, I don't have anything interesting to show you. No, we didn't have any result in the swimming behavior, but it's more interesting when we look at the house pumping behavior. So for this, I also use three main metrics: frequency, which is beats per second that the house do when it's pumping, when the animal is feeding; velocity, which is the speed and distance at which the house expands and contracts; and synchronization of the house, so how synchronized the whole structure is, accounting for left and right side or up and down parts of the house. So for this, if we look into the frequency of the house pumping, we observe differences between the control and noise-exposed animals. And if we look at the videos that I have here, you can see that the control animal is pumping the house in a more rhythmic way, while the noise-exposed animal looks a bit like hyperventilating, like the movement, the frequency is way faster than the control. And so we can say that the rhythmic expansion and contraction of the house is affected by noise. Now for the velocity, we also observe differences in these noise treatments in the maximum velocities, which is how big the house can be expanded. And in this case, the maximum velocities are lower than control. And for the minimum velocities, then we also observe differences. So this means that the animals exposed to noise cannot contract and expand the house at the same extent as the control animals. The speed and distance at which the house is expanding and contracted are affected by noise. And now I would like you to pay attention to the tails of the animals. If you observe here, the control animal has this rhythmic movement, while the noise-exposed animal shows a different pattern, not even the same force of movement. And this is super important for the house expansion and contraction. The tail is the structure in the animal that creates this water flow that will go into the house and will inflate it or contract it. So in this case, we can see that the tail is not really working properly. So for the house pumping synchronization, we just look at the control going more or less stable in mean correlation, while the noise-exposed animals have a different trend, starting from negative correlation to positive. So we could maybe think that there could be a synchronization of the house in response to noise. And we also looked at the correlation between the tail beating and house pumping, which is also interesting. Here we compared maximum tail frequency with minimum house pumping velocity. In control group, we observe a positive correlation, while in noise-exposed group, we observe a negative correlation. This means that the faster the tail is moving, the smaller the house will be contracted in control groups. In the noise, this is not happening, which may indicate that the animals are not contracting the house, or the tail is not doing the force that the control animals do to contract the house. We also observe maximum tail velocity with the maximum house pumping velocity. And we see a high correlation here in the control, while in the noise-exposed animals it's a bit lower. So again, this may indicate that the house is not expanding with the tail movement as the same extent as the control group. So these results give us the question of whether the mechanoreceptors in this animal could be affected by noise. And this animal has two types of mechanoreceptors. These ones are celiac cells that are located in the mouth, and therefore they are open to the water flow and, of course, to vibrations from noise. And also they have Langerhans receptors here in the tail that are also attached to the house, so it makes them more sensible to vibrations. So as this animal is a filter feeder, I realized that it depends completely on the house to feed. So an impairment in these receptors may cause an effect on the feeding rate of this animal and therefore their survival. So lastly, the threshold that we set for this experiment was we observed that even one hour of noise exposure at the lowest volume tested comparing to the background noise that we had, which is not a big difference. We can still see effects of noise in this animal, specifically in the house pumping behavior. So just to conclude, the machine learning approach that I showed you is a novel tool that has been just developed now and that we could use to not only study the noise effects in this organism, but also other kind of stressors. And so Oikoplera dioica is also a great bioindicator of noise, underwater noise pollution. This organism is pelagic and is worldwide spread. So it allows us to use it as a model organism to study underwater noise pollution in other places as well. And also it's very important ecologically and also being part of marine invertebrate eutherstones that are not being studied in these topics. And of course we observe some effects of noise in the house pumping behavior, as I already mentioned. And these effects or these results brings up new questions of the role of the house in the impact of noise in this animal. As we observe these effects in animals that were in the house, we could think that maybe the house could be acting as an amplifier of the effects of noise in the animal. And also during the experiments, I observed that the animals that were exposed to noise always had the house. So I would also think that, I don't know if it could be a coping mechanism that they use to cope with noise. But these are questions that should be further studied. And yeah, so this study gives us this new tool, this new bioindicator, and also contributes to a general big project that is studying only the effects of noise in marine vertebrate eutherstones. And yeah, I just want to thank my lab, my supervisor that are watching, and everyone here for listening. Yeah, thank you.
Thank you, Ingrid. And for the first time, let's go to the jury. Who would like to start?
Okay, yeah, no, I have very few notes even because I was enjoying a lot watching and hearing. Incredible. Really nice presentation. And I don't have so many questions about the scientific point of view. Probably I have a lot of curiosities. For instance, why this animal, but explain somehow, but in any cases.
Yeah, so we chose this animal because it's spread, like it's located in many places. Also because we already had the culture in the lab, so that made it easier. So I was told how to culture it, how to keep them in good conditions, and yeah, it was a sort of resource that we already had. But still, it's a very important in the ecology, marine ecology.
And related with that, what is a eutherstone and why is it important to mention?
The marine invertebrate eutherstones?
Yeah.
So they are animals that are part of the trophic chain, and they are not really studied in these topics, so we don't know what the effects of noise are in them, but if one of them are affected, then the whole, the other organisms.
Is huge, I mean,
Yes.
there are a lot of, but why important to mention eutherstones? It's important?
Yes, it is important. It is a big project that is working in
I
many
mean, you say
jurisdictions.
invertebrates because there are other research with invertebrates, but it's not also a very known
Yeah.
topic.
So basically, this project is already studying different species of eutherstones, so that's why we are focusing in them.
Okay.
And we were labs in Italy, Spain, in different countries that are studying different species, and it's a big consortium.
Okay, so it's because of that, because they use like also somehow something different as well.
Yeah.
The eutherstones, if I'm not wrong, is just that when the embryo started, the mouth is coming from the hand.
Yes, yes.
So it's a big line. Okay, good. My question was specifically for the mechanoreceptors.
Yes.
When I was, but you already mentioned it. So who is like mechanoreceptors, how to understand better that mechanoreceptors. Is it more information than the one you saw us in the presentation?
Yes.
Is well characterized, or
They are well characterized. They are also being still studied, but they are really hard to find. Like we even tried to stain the mechanoreceptors to see if we could see any like damage, like physically damage, and we could not. We didn't achieve to stain them yet. So yeah, I also agree that it's super important to see that, and it will be super interesting to see if they are actually being affected.
because I was looking to the image and I cannot really see the structure. Of course, I don't know, the nerve system of these animals is not complex.
Yeah, I think it's not complex, and that is why they are great model organisms also for neurological studies, like evolution of the neural system.
For sure, it's important, but yeah.
Yes, and yeah, so how they detect these vibrations from noise is because these receptors, that's the function that they have. So they create this action potential in the nervous system, and then they sense these changes in the environment.
Really interesting. And what is the house? What is the material? What is the composition?
It is cellulose and different polymers that yeah.
They
It's like
produce?
gelatinous. Yes,
And they have a
produce.
gland to produce, they have something to...
Yes, they have all the mechanisms to produce them, and they just like create it is called oikoplastic epithelium. So they create it and they leave it around the head of the animal, and then when they leave the house, because that's a voluntary movement that they do, they leave the house when the filters are clogged. So they just swim away, and then they have the ability to produce one, and it is more or less every 4 hours. So they produce a lot of houses in their lifetime. Yes, and they are
It's a waste
also.
of material, come on.
Yeah, I also thought that it has. It's really interesting, and also this house contributes to the carbon flux, because as they get these particles attached, then it goes to the bottom and it stays there. But also they are giving an easier way to like transfer energy from smaller particles to bigger animals like fish larvae and so on. So it's really interesting.
Okay, I think it's... No, just one comment that it was... it's nice that we are always looking for science fiction, to new animals, to new things, and we have strange animals as well. So it's very to enjoy with the animals we have. Sometimes so thank you.
I guess you will have some questions about Ecoflow.
Why?
I remember a few classes from.
Well, we have colleagues, Villefonchemin, right next to here, that Ecoflora was their model animals during their whole life. And they've been working with the SAR Institute.
Okay.
And, but that's not my case. I've been working a little with them, but just from the modeling aspect. So in your very nice presentation, very clear, nice videos. So you had aims. One of them was called machine learning. I understood that in this machine learning there were two parts. One is a kind of hardware part, which was to set up this camera and the lens. And then there was a part which was to set up the labeling of the different movements. So which part were you part of?
Yeah.
And
So
what is your exact contribution?
Yes, so I participated from the creation of the model. So from taking the videos, collecting the videos of the animals, to make this schematic of the body parts and then label them. I got help from one of my colleagues, and we both together, we labeled like 570 frames. So many videos and a lot of hours putting dots. Yeah, so that was how we created the model. Then we tested it 3 times, and yeah, it was when we decided that it wouldn't work for the house yet. But yeah, it would be nice to get it at the end.
So it was a long part of your master thesis?
Yeah, it was, I think, 1 month maybe, or less, because I also learned how to culture the animals, so that also took time.
Yeah,
So it was
because
a-
they are very hard to cultivate, because
Yeah.
if you perturbate them, they will just reject the house.
Yes, that was a very important part. We wanted to record them with the house, and I had to learn how to take them with a pipette without breaking it.
Yes.
That was super hard.
So what is a chemograph? You went too fast on me.
Yes.
I did not understand.
Okay, yes. So chemograph is a tool that is used only with cells, not in this kind of organisms. But basically, it shows us the movement. So if I can reach that part, it gives us this image that shows. Okay, I started with drawing this line in the house without including the tail. That was important. So we would see in the software how the house is expanding and contracting for these two parts, left and right, and then it gives us this image of the movement. And then I analyze it with a different software that through this same movement, it could give me numbers of velocity per
Okay,
time.
because this image was a video.
This image was a video, yes.
Okay, so you could see in this specific part
Yes.
how it was. Okay.
Yes,
right.
yes.
So sound is defined by frequency and intensity.
Yes.
So which one did you use? Are they relevant in regard to what we find in nature and generated by our activities?
Yes, so the sound profile that we used was created with the software. We used frequencies in a pink noise, we call it, like it's like high energy, low frequency noise. So it goes from certain amount of frequency hertz to another, and
How much?
63 to 120 hertz.
Okay.
And that's a low frequency noise that these animals are able to sense. So this is also noise simulating the maritime traffic noise.
So how do you know that they are able to sense this noise?
Because these animals, I think it's because they are like, these kind of animals sense low frequency noise, not high frequency. But
Okay.
I'm not sure like how exactly.
Okay. But so it was knowledge that was a prior knowledge that you.
Yes.
And then for the intensity, you said it was relevant to oceanic intensity that are recorded for the noise.
The decibel level.
The decibel levels.
Yeah.
Oh yes. So we use 3 different treatments of 3 different noise levels, and we measure them with a hydrophone. And so these decibels are just controlled by the volume that we put in the amplifier, but they are in the same range of frequency that we wanted from maritime traffic.
Can you show again just the decibel level? Because
Yes.
I have a follow-up question. It seemed to me that it was super high.
The decibel.
Yeah.
Yeah.
Control 140 decibels is
Yeah.
like higher than any siren of an alarm that we could have in the building. So, is it a control level of sound that we can imagine underwater for
Yes, the thing is that it's underwater, so it gives us higher decibels than if we measure it here. So this was the background noise in the lab where we cultivate the animals, and then we just increase a little bit
Ah,
with
okay.
the amplifier.
It's noisy lab.
Yes.
Sorry, Lars.
Maybe
Last
last
question.
question is in your experimental design. So
Yes.
here I understood that you have tunicate in this tank, that you take it out to measure the individual movement,
Yes.
then you put it back.
Yes.
Okay. What is the effect of so you when you measure the noise exposure experiment? Is it still the noise when you do it, or it's
No, so we expose them to noise, and after we take them out of the beaker and we put them in the camera, so they are not being exposed to noise while recording, and that's the next step that my lab wants to do, but we weren't there yet. So
so does it mean that they keep a memory of the music?
I guess it means that if the mechanoreceptors are affected, they will behave different. So
Okay.
yeah, exactly. So they are still stressed, and it's immediately that we record them.
Okay.
Okay, thank you.
And a couple of more questions, and we're good to go. Coming back on slide 19,
Yes.
please. You showed us, again, fantastic videos, pictures. You took these videos and pictures? Right?
Yes.
Okay. So here you can see really interesting differences between the control and noise 10. It's about dancing. The dance is different. But we are just looking at 2 individuals.
Yes.
Is it something that you observed visually on a lot of individuals, this difference?
Yes. So I have many videos of many different animals, and they have kind of the same pattern of movements.
Okay.
And also we analyzed all these videos. So all the data that we have is from different animals.
Okay, so what we see here between these 2 individuals
It's just one example.
it's only one, but it's a pattern that you see on
Exactly.
others.
Yes.
And maybe to complete on something larger, here you're just looking at the acute effect of noise at the moment, but how would you do to test the effect of noise on the general fitness of the individual, on the capacity to survive, to produce?
So I would think that maybe longer exposure to noise and maybe allow them also to recover. So we had this recovery phase in our experimental design, and I think that could give us also information if whether the effect is like it stays in the animal. So yeah, I would think expose them longer to noise maybe could
And
tell
let
us
them
more.
recover and do it again?
Yeah, exactly. Or see more animals, like more replicates as well. Yeah.
Thank you. I think we're all good to go. Congratulations.
Thank you.
And congratulations everyone, you made it.